whole millets, open and small millets. I also heartily welcome Dr. R. Madhusudana, a dynamic coordinator, project coordinator, Superman Millets, whose contributions and dedication have been vital in the organization of this combined annual group meeting on Millets, Superman and small millets. I also extend warm welcome to Dr. V. Sumati, Associate Director, newly joined, and her colleagues from the Agricultural Research Station, Tirupati, and also the principal, Dr. Ramaji, who are very friendly and also very cooperative in organizing this event. So furthermore, I welcome all the delegates involved in the research and development of millets, sorghum and small millets from various parts of this country. And your participation in reviewing the results and formulating the effective technical program the attention is invaluable to this combined annual group meeting. I also extend a warm welcome to the press, electronic media, including our radio, who play a very important role in the technology transfer. I welcome the teachers, students, on this class providers, Tirupati, scientists from Southern Jew, retired scientists and advisors from ICR and Government of India organizations. Further, I welcome the staff of ICRI and Hyderabad, ICRI on table minutes, sorghum and small millets, and chairpersons and co-chairpersons of the various technical sessions. Thank you for all joining us today for the inaugural session on the combined annual group meeting on table minutes, sorghum and small millets. Colleagues, the challenges are very and unprecedented. There is a great potential developing improved varieties of millets, sorghum and small millets with high resilience to various biotic and abiotic stresses, strengthening research, developing new varieties, enhancing processing and value addition through improved technologies and expanding exports can significantly raise farmers' income by increasing the farm productivity. So this combined annual group meeting is a fitting platform for fostering continued cooperation among all the stakeholders. It enables us to focus on our efforts in translating laboratory outcomes into field applications directly benefiting the farmers. Additionally, it is crucial for the next generation of the researchers from SUs and ICR institutions to stay updated with cutting-edge technologies. So in this direction, I just would like to enlighten the annual contribution with these three medicals in just 1 minute, sir. Agro has developed so far 76 million varieties, including popular like NTG, that is Johanna, the sorghum, and very popular not only in the state of Andhra Pradesh, in other states in India. And NTG 50 is also very popular. The biofortified composite variety ABB 04, which was released very recently in Bhadra, and also multiple varieties in ragi such as Megavati, Swarnamugi, Goshani, Vakula, Tirumala, Gautami, and CFME 4. For foxtail millet also, notable contributions include Suryanandi SA 3156, SA 3085, Garuda Renadu, and Mahanandi from a recent research station. So our research efforts also include registering genetic stocks with 16 in figure millet, 1 in millet, 1 in little millet, 5 in foxtail millet. You know very well, sir, the Anglo 4 active sectors in sorghum, pear millet, finger millet and foxtail millet, and 9 research stations across the state of Andhra Pradesh are involved in millet research and promotion, supported by 19 scientists and KVKs and DOT centers, technology transfer centers, also play key role in introducing our new varieties and getting popularized. Conducting FLDs, OFTs and tribal sub-plan and etc. are multi-based entrepreneurship programs, and Anglo also conducts an annual millet seed production program to distribute improved varieties. Overall, Anglo is actively promoting millet consumption, production and marketing. Sir, I assure you, sir, I am also happy to inform you there are many FPOs, especially in north coastal districts. They are very actively promoted by our head of our IIMR, the Tara Sachet Madam, Srinivas Babu, who are very actively involved in promoting the millets through FPOs. Very happy situation, sir. That's done in harmonically. Sir, if possible, if any time chance comes, North Coastal District is very important for millets, and the FPOs are doing excellent work, not only in promotion, production, also the byproducts utilization, valuation, etc. So we are doing many multiple programs to promote our millets, sir. One such program I just tell you, sir, it's not just the varieties alone will contribute for the increased production. It is the management practices. It is the combined management practices play a major role. For example, the ragi is very important crop in North Coastal District, Andhra Pradesh. So the ragi, because of the cultivation expenses, cost of cultivation, labor problems, the ragi is getting down. So to combat that, what we have educated like paddy, ragi, sequential crop. A relay crop like paddy pulse crop, we are now concentrating on that. As a result of ragi seed will be sown just before harvest of the paddy, and a lot of water is saved, and like that we are doing some new such management processes. We are focusing at our actual engineering university. Thank you very much, sir. So I trust that over the next 2 days you will find the proceedings of this group meeting to be engaged and productive into the fruitful results. Thank you very much once again, inviting you all and all of you to this 2-day session. Thank you very much.
Thank you, Dr. Satyanarayana sir, for your elaborate opening remarks and also highlighting the research achievements of ANGO. Next on the line is the presentation of research highlights. For this, the initial request Director ICMIUR and Project Coordinator Dr. Singh Prabhasakirti to present the research highlights in 1 minute.
A very good morning to you all. Most respected Deputy Director General Crop Science Dr. T.R. Sharmaji, respected Vice Chancellor Dr. Sharda Jayalakshmi Madam, Assistant Director General Food and Product Crops Dr. S.K. Pradhanji, Director Research Dr. Satyanarayana Garu, and other dignitaries on the dais and off the dais, my fellow colleagues from different ASRP centers. Let me present before you the research highlights and annual progress report of All India Coordinated Research Project on Pearl Millet. Next one, please. So just before going to the overall outcomes, I just want to take you around for 1 minute about the pearl millet in the scenario, Indian scenario, and how it is about. It is the fourth important food crop in India after rice, wheat, and beans, occupying at present 7.37 million hectares with a production of 10.67 million tons and productivity of 1449 kg per hectare. And the major contributing states of pearl millet in India are Rajasthan, Maharashtra, Gujarat, UP, and Haryana. Next one. And the most common challenges faced by pearl millet are high temperature and low moisture during seedling stage establishment, low soil fertility, white stresses like blast, downy mildews, mud, turbot, and rust, poor shelf life of pearl millet flower. Next one. And if you see whatever experiments we are conducting in this AICF and pearl millet, we are doing based on the agroclimatic zone or grouping of those areas which are suitable for pearl millet. So we have 3 major agroclimatic zones. Then adding to that, we have one summer agroecology, that is 4. We have A1 zone, which is in the northwestern part of the country, comprising most of the Rajasthan, then parts of Gujarat and a little part of Haryana. Then A zone, which is containing North Indian states like UP, then parts of Madhya Pradesh, Gujarat, and others. And then B zone, comprising of the southern states of India like Maharashtra, Karnataka, Andhra, Telangana, Tamil Nadu, etc. And in the summer category, we have the area in northern Gujarat and southern Rajasthan, where farmers take pearl millet after harvesting of potato. Next one. If you see the trends in pearl millet area production and productivity, despite the decrease in area from about 10.5 million hectares to the present state of 7.37 million hectares, the production is constantly increasing. You can see right now the production is about 10.57 million tons, and the upper one, the white, the yellow colored line, that is showing a continuous improvement. And right now, in this year, the productivity is about 1447 kg per hectare. And it started, take from 1950s, That time the productivity was only 300 kg per hectare. And now we have reached to 1447 kg per hectare. Next one. And this all could happen because we are breeding based on the requirements two different agroclimatic zones. So A1 zone, as I mentioned, we need short duration hybrids and populations. And when B zone, we require and target medium and late maturity types. A1 zone also the same type. And in the summer pearl millet ecology, we go for those hybrids and varieties, majorly these hybrids which mature around 80 plus days. Next one. And in the last year, if we look into, last year there were about 10 hybrids of pearl millet which have been notified and one variety which has been notified. In A1 zone, we have two hybrids, GHB 1294 and FPMH 42, and one GHB 538 improved, that is an EVB, which have been released for cultivation. In A zone, we have HBH 191294 that was released for cultivation. And in summer growing areas, two hybrids 86M22 and PB187 have been released. And there are other state releases coming from Gujarat, GHB 1351, Pro coming from Haryana, and PHB 1625 coming from Telangana. These have been released for cultivation. Next one. This year, in 2023, we have about 125 new experimental hybrids which have been tested in 14 different trials at 62 different test locations. These are the different trials you can see. Those which have been highlighted in red color, those show that is the maximum yield potential we are able to get in those fruits. Maximum we can see high amount of productivity and high yield in case of late maturity types and also in the summer hybrid trials. Next one. And these are the entries which have been promoted to different trials. Next one. This is for A zone, the previous one was for A1. Next one. And this is for B zone. And this is for again B zone summer trials. Next. So in all these pearl millet improvement trials, the hybrids which have been generated and tested, a total of 71 A lines by name and 77 R lines by name have been used in generating these hybrids. And apart from testing for only yield and other agronomic parameters, you all are aware that pearl millet is the first crop to introduce the benchmarking level of iron 42 ppm and zinc 32 ppm. And in this process, we are testing all the hybrids which are having high yield, and then we are testing for iron and zinc on multi-location and replicated basis. So this time what we have observed is the iron content ranged from 41 to 99 ppm and zinc ranged from 27 to 60 ppm. And not only these, we also test for protein content and fat content. Next one. In the last 23-24, the breeder seed production we have taken up for 54 parental lines and 7 OPVs. A total of 26.953 quintals of breeder seed was produced against a target of 10.978 quintals. And we are also involved in dust testing. About 14 candidate varieties and different varieties have been tested along with the local checks at both Mandor and Dhule. And also a separate dust character study trial was conducted with 34 parental lines so as to generate the data and maintain the database. Next one. Under crop improvement, we also have ICR Ecoside partnership trials. A total of 126 R lines and 90 B lines were assessed in 5 different trials at 12 locations. One of the trial is the combined trial, that is elite joint biofortification trial, wherein we have tested 50 entries across 7 locations, and we could get some lines which are having a high amount of iron compared to the checks. Similarly for zinc also. Next one. And then the CRP biofortification trial, which is funded from ICR, we have 33 entries tested at 6 locations, and we could get iron content ranging from 47 to 88 ppm and 30 to 57 ppm for zinc. Next one. We have a small lab at PC unit, wherein Dr. Supriya is taking up molecular breeding trials, experiments. We have 4 experiments like validation of markers in pearl millet and molecular characterization of germplasm of pearl millet for the various traits. And this is the information regarding number of entries tested and number of markers used, and then polymorphism and other things. Next one, please. In agronomy, we have conducted about 6 experiments, and these are the different titles of the experiments, for mainly focusing on moisture conservation, nutrition management, crop management, and then natural farming and resource conservation technology. Next one. Under the first one, that is PM81, wherein we try to come up with response of our advanced entries to different nitrogen levels, we found that 90 kg nitrogen per hectare was showing good amount of yield, and then enhancing biofortified non-biofortified pearl millet hybrids, then we could see that based on 3 years of data, we could see that the micronutrient treatment of zinc sulfate plus ferrous sulfate plus 0.2% works. There in zone A1 we got 32% superior yield. Then in zone A we got 29.5% superior yield and in grain, and stover 13.3%. Zone B we got 32.4% superiority in the grain A. Next one. In the experiment related to contribution of various production factors to yield and other economics, we could see that in zone A1, because there was no irrigation, there was a reduction of 64.1% in the yield. Whereas in zone A, we could see that when there was not application of recommended dose of fertilizer, we could see 45.3% reduction in yield. Similarly, in zone B also 48.3% reduction when RDF was not applied. Next one. Next one, madam. When we are seeing the experiment kind of related productivity of pearl millet related to cropping sequences for influenced by organic or natural farming, this is a second year of experiment. In zone A1 we could see that when the yield is improved by 50.9% and 55.5% over the recommended dose of fertilizer, then cow-based formulations were used. Similar kind of observation also we found in zone A and zone B. Next one. Apart from agronomical experiments, we also take up frontline demonstrations. This year we were allotted about 666.8 hectares of frontline demonstrations. We have conducted out of 1000 hectares, and in Kharif we conducted for about 601.8 hectares, and in summer we conducted for 65 hectares. We can see that the improvement is in zone A1 we could see 19.8 to 45.8% improvement with the improved technologies. I am sorry because of some irritation. In zone A we got 10.4 to 24.7% improvement with the technologies. In zone B it ranges from 13.7 to 28.1, and in summer ecology it is from 6.7 to 23.6%. Next one. We have conducted 4 physiology trials. We have 3 locations, Mandor, Jaipur and Jamnagar, but due to the transfer of the physiologist from Mandor, the experiments were not conducted properly, so that data was not considered here. And in heat stress characterization, the following inlets were found to be performing better in high temperature condition in summer. And similarly for the drought tolerance experiment, the parental lines, inlet lines BBI to 20401 and others are showing better performance. And photosynthetic partitioning and recombination, we found that the assimilates at emphasis and maturity distribution pattern is more good. And in the 4th experiment for drought tolerance, we found that the following traits like number of leaf, number of tillers, and these contributed significantly for drought tolerance. Next one. In pathology, we have about 9 experiments conducted in different zones, in zone A1, A and B, in different locations. Next one. So if you look at the gist of the things, in 5 trials, these number of entries have been tested, and we could find highly resistant entries for downy mildew, glass, rust, smut and ergot. And these will be used in further breeding also, because when we go back to the pedigree, those particular parental lines will be again used for coming up with new hybrids. Next one. In entomology experiments, we have about 8 experiments conducted in different locations at different centers. Next one. In the first experiment, which is regarding the screening of initial test entries, so we could find Jamnagar and Fatehpur Shekhawati's shoot fly incidents In stem borer instance was more in Jamnagar and Fatehpur Shekhawati. Helicobacter armigera again at Fatehpur Shekhawati and Jamnagar. Grey weevil incidence also more in Fatehpur Shekhawati. Leaf roller incidence again at Fatehpur Shekhawati. Next one. And in advanced trial screening, shoot fly was more in case of again Jamnagar and Fatehpur Shekhawati. We could find more incidence of shoot fly, stem borer, helicobacter, and also grey weevil. Next one. In another experiment on monitoring of insect pests and resuscitation, and also we have another experiment on monitoring of insect pests in farmers' fields. So these are the different insect pests we could find. Next one. In use of chemicals for testing against helicobacter, we could find the spray of imamectin benzoate was effective to control helicobacter. And survey of insect pests in summer ecology also we could find that shoot fly, stem borer, grey weevil and ladybird beetle were seen in significant number. And then in monitoring of fall armyworm, this is done at 5 locations. We could see based on the catching of those moths in the traps, it was more in Jamnagar. Next one. In the last one, that is the storage pest trial, we could see that incidence of Tribolium was more at Jamnagar than Rhizopatha dominica in case of Anand. Anantapur again Rhizopatha dominica. Mandor and there also we have seen significant incidence of Rhizopatha. Next one. And in the last year our State Agriculture University ASRP centers, they have done some MOUs to promote seed production and penetration of the newly developed hybrids. HISAR has done one MOU with the US Agri Seed Agency, Indo-US Agri Seed Agency for HHB 299. SKMU Jogne they have done for 223, RHB 223 Bajra hybrid. Then TNAU has done one MOU with FPO in Dingam Dania Farmers Producer Limited to promote millet value addition technology and other things. Next one. Last year being International Year of Millets, a number of trainings, conferences, workshops, seminars were organized by all the ASRP centers. Since time is limited, I just try to pick up 1 or 2 photos from different centers. So a number of events are organized at PC Unit, Durgapura, Hisar, Anantapur, Ludhiana, Dhule, then Aurangabad. Publications, we have about 64 journals, research articles. If you look into more than 10, we have 5, 7 to 10 we have 7, 6 to 7 translating 2, and less than 6 we have more. So we need to increase the publication from less than 6 translating to more than 7 minimum. And these are the different publications like books, book chapters, technical bulletins and popular articles. Next one please. And that's all for from Pearl Millets side. Thank you for giving me this opportunity and for your kind attention. Eat millets and stay healthy. Thank you.
Thank you so much, Satyavati ma'am. Now, we humbly request Dr. R. Marsudna sir, Project Coordinator Sorghum and Millets, to give the presentation of Research Highlights 23-24 in Sorghum and Small Millets.
Very good morning, respected DDG sir, Dr. T.R. Sharma, respected Vice Chancellor Sharda Jayalakshmi Madam, and guest of honor Dr. S.K. Pradhan sir, PDC, Director Madam, Dr. Prada Sakavati, Dr. M.E. Rona, Dr. P.V. Satyanarayana, and also Dr. Sumati Madam. Some participants from directors from various ICR institutes and also my other participants. Welcome once again to this AGM. So I would like to present the progress of research and research highlights and sorghum and small millets for the year 23-24. So next slide. So for this ASRP, so we have 8 crops starting with sorghum, finger millet, foxtail millet, brown top, kodo millet, little millet, prosa millet, and And this is the mandate that we need to develop hybrids, varieties, and also trait-specific generic stocks. And also then we need to develop notification, testing for coordinated trials. Then we develop integrated production technologies, and also we undertake participate in this reducible production, FLDs, TSP, CSP, and NER programs. Next. Coming to these crops, so you know that you can see from this table that of sorghum, finger millet, and groundnut, they are originated from Africa, whereas Buda, little millet, they are very specific to Indian context, that is they are originated in India. Whereas Bana millet and Prosa millet, Bana millet and is actually Bana millet and Prosa millet, they are also have two centers of origin. So India is very rich in millets because of its origin and also the diversity. And most of these crops actually mature between they are early in maturity, and they by maximum 120 days they go for final maturity. Next. So these are the active centers for sorghum. So we have around 18 sorghum centers. We can see from this and around 13 centers on small millet. You can see from this graph that mostly sorghum is actually concentrated most in the western part of Karnataka, this are India, whereas the small millets, they start from this southern region and also they go along with the eastern region. So we have very specific centers of the sorghum which are focusing only on forest sorghums like Hisar, Panagar and Ludhiana. We do have centers in Maharashtra, Karnataka where focus is on grain sorghum. And we do have centers which are focusing on this grain sorghum, rabi sorghum, and also sweet sorghum. So next. So coming to the staff positions, sir, we have around 94 scientific position with 92 technical support, of which we have with position 74 and 57, around 20 and 35 positions are vacant under the scheme. And thanks to council that we got last year around 2,274 lakhs of budget, and of which we completed almost 100% we spent on the activities of this agri program. Next. So these are the trials conducted in 23-24 across sorghum and small millets under breeding, pathology, entomology, agronomy and physiology. So out of which around 823 total trial sets were sent to around 176 number of centers. Next. Coming to the achievements under breeding for sorghum and small millets. Next, madam. So during this 23-24 sir, we achieved this 27 varieties were notified under CVRC. Out of which 11 were central and 16 were state. For sorghum we had 13 entries released, of which 4 were central varieties and 9 were state varieties. Similarly for small varieties we had 14 varieties released, out of which 7 were central and 7 were small varieties, state varieties. Next. We also registered around 13 novel genetic stocks during this year, of which 7 were sorghum for earliness, high iron and zinc, cruda and leaf traits, whereas for finger millet we registered 6 for earliness, banded life trait, bleed, and neck and finger blast resistance, high fingers, non-virgin and long finger length. Next. Coming to this year's VAC proposals, we have around 20 VAC proposals, of which 13 are for sorghum and 7 are for small millets for various category of kharif sorghum, sweet sorghum, porous sorghum and specialty sorghums. Whereas small millets will have proposals for finger millet, foxtail millet, prosa millet, little millet and brown millet from various equip centers. Next. So this is the first national yellow sorghum variety, that is CSV 60 yellow. So this has been released, notified last year with and this year's iron and zinc have generated around 32 ppm of iron and 25 ppm of zinc. Next. So these are the various coordinated trials conducted under breeding. So of which for kharif we had around 10 trials tested under June 1, June 2 and June 3, whereas rabi sorghum we have 4 trials on advanced trials and IDTs and ASTs, specialties and also HODA trials. So we could see that from this thus we have the genotypes which can go even up to 5 tons per hectare the potentiality which has been seen in ABHT and also in IHT. So we do have some genetic highly potential hybrids in kharif sorghum. And in rabi sorghum for the first time I think we have a hybrid which is actually reaching around 3,500 kg per hectare. So next. So coming to this kharif, we have one hybrid, that is SPH 1974, tested over 3 years, and it has been found to be good across all the zones, that is Zone 1, Zone 2, Zone 3, and this is being proposed for this varietal rule this year. And we also have this 2871 variety, which is very good in Zone 1, but it's on with checks in Zone 2, Zone 3. Next. So these are the Rabi sorghum varieties which have completed 3 years of testing. So and we have SPV 29 9, which are around 4 to 5% higher green yield compared to the CSP 29, and also it is very bold. Rabi sorghum is one of the important trait for Rabi sorghum for farmers' acceptance is the seed size. So 3.54 is very good seed size we have observed in this SPV 29-19. Next. Coming to this 4S sorghum, we also have this 4S program. So performance of single cut forages, we have this SPV 2878 was found to be very good in both Zone 1 and Zone 2 and also under all India. So this has given a positive around 5 to 70% over the check in for dry barley. So this is also there for this proposal. We have another variety 1881 is good for Zone 1 and whereas 1884 is good in Zone 2. Next. So we have the other component is multi-cut sorghum, wherein this SPH 1998 was found to be very good in Zone 1 and across India with a total of 5.4% improvement over the existing check, that is 24 mm. Next. Coming to this sweet sorghum, last year we released 58 SS, sweet sorghum with a bricks of around 17%. K yield of 53 tons per hectare, and also juice yield is around 16 kiloliters per hectare. So there were many sweet sorghum varieties were tested in this trial last year. So they were found to be very good for the stalk yield and juice yield. Some are 1383, 1382 they have listed here. So they are found to be very good. Next. For this computed bricks and computed ethanol yield are other important traits for sweet sorghum. There also we have very good lines which are improved over the existing checks. Next. So SPV 2819 was tested over 3 years. So this has been found to be very good in its traits like fresh target is more than 15% superiority, 19% superiority for juice yield. It's on par with the check for bricks, and also this ethanol yield is around 18% superiority. So this is one variety we are finding very good for the sweet sorghum. Next. So Agronomy, coming to Agronomy, we have one recommendations of this year. So this is the boron fertilization on Rabi sorghum, which has actually given around 16% improvement over the existing control. And because this improvement has happened because of the enhanced fertility and also seed size. Next. So these are other agronomic experiments where irrigation has improved Rabi sorghum productivity with around 28%. And the spray of nano-urea at panicle initiation stage also impacted in grain productivity by around 20 to 33%. Next. So application of FIM and also this vermicompost was found to be very superior to no application of fertilizers and biofertilizers respectively. And various and biofertilizers were found to be the best treatment for improving the grain yield. Next. This is on the organic. Coming to this summer sorghum, because summer sorghum is becoming very popular in the recent times, so we also conducted summer sorghum experiments wherein we found the VJ 296 was found to be on par with this kharif hybrids, and these experiments are being continued. Next. So under climate change coping experiments also we found that this advancing the time of planting by 2 weeks and has found to be very, and with the lower population size also it is found to be very productive. So this experiment will also be continued. Next. Coming to entomology, we conducted all the experiments mandated for these breeding trials, both in kharif and rabi, and also we undertook experiments on shoot pest nursery and also this pre-breeding derivatives of F6 for evaluation and shoot resistance and stem bore resistance. Next. So grain sorghum, in the grain sorghum categories, we found that none of our hybrids are inferior to the susceptible check, and many were found to be on par with resistant check, that is IS 2205, both for this stem borer. Next. For shoot fly, you can see that this pre-breeding derivatives, pre-breeding activities we started some years back, and we are finding the results now. So we can see that some of the derivatives in F6 which were found to be resistant for shoot fly and also stem borer. So some are common also. So there are multiple pest resistance they are having. Next. So this is for identifying the shoot fly complex. Earlier we thought that only Atharagona saccata is there in sorghum, but we are finding that in addition to that there are many species. They are there. However, Atharagona saccata is predominant. In addition to that, we have Atharagona oryza, simplex, and also Pilla. Many cases they are causing damage to the sorghum. Next. So this is another recommendation from entomology, sir. This is actually against palm armyworm, where the application of chlorantraniliprole at 0.3 ml per liter, followed with this entomopagapathism, that is application of metrorrhizum relay, was found to be very good in controlling the palm armyworm and also in improving the productivity. Next. So yield loss, as per your suggestion, sir, this yield loss assessment experiments are also taken in the last 3 years, and we found that avoidable green moderate yield. Actually, this was conducted against foliar diseases. Forage is very important component of nowadays in sorghum. So we took against this anthracnose disease, which is very important in foliar sorghum. Avoidable loss is around 12 to 14%, and whereas avoidable seed loss is around 20 to 24%. So new molecules for ergot management was also worked out. So this is very important in terms of this seed production crops, where ergot is becoming menace. So there also they identified 3 promising populations, TE4, T5 and T2 are found to be very significant in reducing the ergot and improving the productivity. Next. Frontline demonstrations in sorghum are conducted over various centers where we across various farmers fields, wherein 47% of grain productive is a gap and 43 for storage was found to be after this gap. Next. FLDs were also conducted in Rabi sorghum, where we have 34% grain and as well as sugar in the advantage of the local seeds. Next. This is coming to the some small millets. So these are the summary of trials, where various trials, around 125 entries were tested, and the experiments are conducted at around 180 locations, and we have 95% of the success rate is in conducting these experiments. Next. So these are the best performing entries for over 3 years for finger millet, foxtail millet, rosa millet, and bronta millet. So these have been proposed for this VAC today. Next. Small millets, coming to small millet entomology, earlier this sorghum, the shoot fly was thought to be present only in sorghum, but we can see that the shoot fly is equally damaging the millet crops, of which little millet and closer millet are more susceptible. Besides that, we are also seeing that there are many other species of sorghum. This shoot fly is also, they are damaging small millets also. Next. Promising entries have been identified for various important insect pests in small millets, like finger millet against aphids and caterpillars, egg caterpillars, and also stem borers. Besides that, many entries were also identified for shoot fly resistance against in the kodo millet, little millet, rosa millet, foxtail millet, and walnut millet, and bronta millet. Next. So coming to pathology, in addition to this routine these activities like surveillance, evaluation, and disease management, we do have some genomic activities being done at small millets pathology program. Next. So these are the various centralized pathology trials that have been conducted, where this mandated breeding trials will be there, yield loss assessment trials are there, management trials are there, and also cross-innovation pathology activities are basic activities. Next. So these are various promising entries performed against various diseases. Next. Similarly for other diseases in different crops, species. Next. Same thing against other species, other diseases. Next. So again the same thing against different diseases. Coming to this yield loss assessment, again sir, we took this experiment in small millets also. So the disease considered one is blinded leaf blight, and you can see that this yield loss, available yield loss is almost, it is touching 60% in these crops, because various these finger millet, foxtail millet, little millet, kodo millet, almond, and those are there. Next. So under agronomic trials, these are the various agronomic trials they have conducted. So pre-released evaluation of pre-released genotypes for fertility levels, nutrient management, weed management, cancer conservation experiments, millet-based cropping systems, evaluation of producibility and the performance of economic evaluation of farm machinery also we have taken. Next. Before one. Yeah, that was about this FLDs where this small levels also we conducted FLDs and 20 to 30% yield gap was found in most of the varieties. This FLDs. Coming to this breeder seed production, we are actively participating with the help of ESLP centers. We can happily say that around 48% of the surplus of production is there with this breeder seed production for various crops, so starting with sorghum, pingan millet, foster millet, wheat millet, kola millet and barley millet, millet. Next. So under our approach, we also undertook this PSP, SCSP and GSP activities. All the centers were very actively participated in these programs in propagating the millets and for tribal people and also SCSP population and also in NER regions. Next. Coming to this pre-breeding activities in sorghum and small millets, as per our advice, we started with sorghum in the last 3-4 years. We do have now very excellent derivatives done for this under various approved programs, centers under the guidance of IMR for grain mold, leaf diseases, rust and downy mildew. We have for shoot boll, stem borer, midge, aphids and shoot bug, brown coat and salt organs also. We do have some process made for this maturity, fertility restoration genes, alternate exploiting alternative cytoplasms and also biofortification. Next. We do have some inter-institutional breeding activities where we share the efforts and also segregating material with other athlete centers like a shuttle breeding concept. The other ways we are focusing more on this multiple tailor parent process like magic approaches. We are concentrating to develop hybrids for rabbit sorghum where none is there at present. And we share trade donors and germplasm lines across the ASRP centers. We are also initiating some genomics and mass programs for starting with sorghum. So we do have mid-density SNP panels available with the CGR system. So we are going to exploit it for all the genomic activities. We do have now association mapping panel and the GS panel is also formulated and master seated for stage green QTS, especially 3A and 3B is also underway. Next. Coming to these biofortification varieties, compared to this polymerate, the millets do have lesser levels of this iron and zinc. So over the 3 years of experiments, we have come up with some baseline levels in millets like sorghum. So 22 to 25 is there, zinc is around 15 to 18% ppm, protein is around 9 to 10%. Keeping in this low levels of iron and zinc, so we have formulated that anything over than 30 ppm for iron and 20 ppm for zinc and protein more than 10% should be considered as a biofortified variety for sorghum. So that we will be treating under special category like specialty sorghums. Similarly, we have small millets and also pure millet. Next. So we do have some opportunities for propagating the sorghum and small millets in other areas of non-traditional areas. Like now summer sorghum is becoming very popular in Karnataka and Maharashtra. Similarly in other states also. So we have formulated one experiment, one trial on this summer sorghum this year, sir. Next. So we do have, this is a lone, rice palace is grown in sorghum. So this is also being continued. But what we are going to do is we are going to breed for the plant which is actually required for this rice palace, like medium height with more grains, high input responsive, suitable for mechanization, very important, and yield potential we are expecting to be more than 6 to 7 tons per hectare. Next. So timber millet is another important, sir. This is being getting very popular in summer. River bed and irrigated irrigations. Where Chhattisgarh and Karnataka centers of Maharashtra and Jagdalpur they are actively participating in these programs, where you can see that cultivation is being done on the river beds and also under limited irrigations, where they are getting almost 35 to 40 quintals per hectare productivity. Next. So this is another important is intercropping the timber millet in sugar cane. That is also happening in Maharashtra and even in Karnataka. So these also we are going to initiate some experiments on this on the agronomy. Next. So there are some new initiatives in sorghum. We are going to do this short nature erect with hybrids for high density planting, red sorghums for export market, assessment of genetic years through aerodynamics conducting, and specialty sorghums for Huda, pop and color sorghums, and also place. This segment is not exploited, so we are going to concentrate only on that so that we can add value to this crop. We will have students guided for MSc and PhD at various Africa centers in addition to IMA. We have very good publications done by Africa centers, starting with food chemistry, frontiers in plant science, frontiers in nutrition, and many papers in crop science, all from various Africa centers. Besides that, we have plenty of books, publications, and participation in conferences, etc. Next. So coming to this last slide, sir, this is actually the crops and what are the target rates. We are looking at kharif sorghums. We have hybrids and varieties. The targets will be for wider adaptation so that it can be cultivated in soil, kharif, rabi, and even summer. Industrial users is one trade. This we are looking for this kharif because it doesn't have any food value in kharif. So therefore, we are looking for this industrial applications, biofortification, grain mode and shoot time becomes the important traits in kharif. Similarly, in rabi, we don't have any hybrids. So but therefore, genetic diversification in the UV and R background is very important. Terminal growth is as it is there. Cold tolerance is very much required in the recent times that we are focusing now. Sweet sorghum, high biomass, we have biofair, porous, stem, bora resistance, and bora and juice quality. Under porous sorghum, we have the traits like high productivity, porous quality, shoot resistance, and floral disease. Small millets, we will have this floral biology and crossing techniques needs to be part of all the centers. And recombination, mutation breeding, and new sterility systems, at least in timber millet and foster millet, we need to have it so that hybrids can come. Last, BB and heat tolerance in small millets, and shoot fly is very important in little foxtail and also closer millet. Next. So coming to this, some of the PMC observations last year, and we have already supplied this from our side, but to say all the whatever the observations were made and PMC committee, we have taken care of all those, both in millets, sorghum and small millets. All the crops we have taken care, sir. Next. Similarly for small millets also we have taken care of all the activities which are already there, but we still will be emphasizing some of those. Next. Okay, with that I think, thank you very much for thank you.
Thank you, Madhusudan sir, for your presentation on research highlights in sorghum and small millets. Moving on in this inaugural session, now we request our guest of honor, ADG Field and Flower Crops, Dr. S.K. Pradhan, for his inaugural address.
Very good morning to all. Honorable DDG Crop Science ICAR, Chief Guest of this combined annual group meet of millets, distinguished guest, Honorable Vice Chancellor of ANGRO, the prestigious university of this region, who was Director of Millet Institute, IIMR, Hyderabad, Dr. Taha Satyavati, Director of Research, Dr. Satyanarayana, ANGRO, with us, Dr. Ramana, Associate Dean, Madam Sumati, and Dr. Madhusudan, Project Coordinator. Also distinguished participant I find here, Director of Seed Mall, Director of DPK Salmoda, and our ACRIP advisory members. Also here, all the faculties, local organizing committees, chairman, committees, organizing secretary, participants from various Millets center of the country, being with us, all the volunteering students and faculty member of this college, press and media. A very good morning to all. You know, everybody, millet is very important last few years. It is already known, and we have already celebrated the last year the International Year of Millet. Before that, also we have celebrated the National Year of Millet 2018. So why we are celebrating, you know, the very concern is that the millets are very nutritious and nutritional value as well as the iron, zinc, antioxidant content and other properties of these millet are very good. But surprisingly the production and productivity of these are very low. Also the main concern is that its area is declining day by day. As you were listening to the presentation of Madam Satyavati, even if we were thinking this palm millet will be stagnating, that is also slightly declining. It's very concern. Similarly, sorghum, you know, the kharif area is sharply declining, as well as, you know, the small millets, productivity area, everything is, we are concerned, we should see that. You know, these are very much nutritious, very important food grains, sometimes used as also nutraceuticals. So therefore, we need to give more focus about this. For creating awareness, actually we celebrated this International Year of Millets as well as this National Year of Millet in 2018. So this is, you know, this is an effort just to create awareness. And I just remind you that these were cultivated in very large area, mainly about something 35 million hectare. If you go back to 70s, the area coverage was 35, about more than that. But now what is the area? You see, it's less than 15 million hectare. But we are happy to see that the productivity is increasing. That is the good part of it. So the government, everybody, NGO and other organizations, everybody they are trying to create awareness, importance of these crops. So very lot of things, good things are there in the millets, you know. So our country actually, we achieved food grain sufficiency, but we did not get this our nutritional sufficiency. So government planning is that this along with the food grains means major cereals, say for example rice. Along with rice, each person's plate, it should be rice or wheat, it should be also millet should find a place in the plate. That should be in the common people's, poor people's plate should be place in the plate, so that this nutritional deficiency, this importance of these millets, the people can, instead of this nutritional requirement, they can get from the millets. That is the main target, but the concern is that there is a rapid decline in area, so we have to think that how much then we need. What is our requirement? We are giving so much of efforts. You know, now we are producing about 17 million tons, like that. And we are able to manage our requirement, and a little bit of it we are able to export also, like 1.6 million tons of lakh tons of millets we are able to export, very less. This is very meager quantity. But the thing is that we need the way we are campaigning, creating awareness and everything. Government is targeting to increase this production. By the end of the Amrit Kal, you know, by the end when we are going to be a developed nation. Since this food sufficiency along with nutritional sufficiency government wants. So therefore we need to increase our current level of production and productivity. Also we want to increase the area of these millets. So therefore, how much we need, what is the requirement? That is the one thing of concern. One estimate we are doing, if we will be at the current level, we will be thinking. So by 2047, by the end of the target period, we may need about 20 million tons here like that, just current level. But the way the government is thinking and fulfilling the nutritional requirement and other target, and our export, creation of awareness internationally, nationally. So thinking of point to constant increase, that is demand elasticity. So we need about 40 million tons by 2047. So at least near that we need, so that we will be getting, we will be exporting and we will be able to self-sufficient in nutritional security also. So how to do that? This is a challenge for us. It's from 17-18 million tons to reaching to 35 to 40 million tons within coming 15 years. So it's a really great challenge. And you know, the millet crops, this improvement is very less as compared to rice, wheat, maize. The millets are not improved so much. So less work, less improvement work is taken care. And therefore a lot of focus emphasis is being given for improvement of millet. So in that case, you know, so many traits, so many mechanisms yet to be studied, to be known. Those mechanisms, unless we understand the genetics of resistance, the genetics of all quality parameters, we are telling quality traits, like low glycemic index, then antioxidant content, rich iron, rich zinc, and so many nutritional things. Unless we know the genomic region here where it is controlled, we will not be effectively breed the variety. We will be, instead, we will be adding up so much of brand, so that instead of the desirable character, we will be adding up the undesirable ones. So first thing is we need to understand the mechanism. Unless we do that, it is difficult to improve the crop. You know, we have to improve the crop. Many of these as Dr. Madhusudan was showing, and these are, well, to many of these millet are home to India only. So we have to improve that. The other people, other have not, they will not be thinking our crops. Therefore, last few years we are giving emphasis to study simultaneously this yield improvement. Simultaneously we want to study the genetics mechanism also along with using our partnership, so that the phenotyping work will be taken up at the centers, locations, targeting few of the centers where the facility phenotyping are there, and using our headquarters where they will be doing the genotyping. So in that way we will be able to understand almost all the important traits which are still neglected for millets. We will be studying those mechanisms. Besides, wherever already reported, there are also many reported gene skippings in this, also showing multiple resistance donors are available. So in those cases also we are emphasizing how to put together, not stepwise transfer one or two traits, so that in the coming 15 years we will not be able to achieve our target. So we are now focusing how to simultaneously transfer together multiple traits, not only vitamin, zinc, iron, yield traits. We will be targeting more, so that also Dr. Madhusudan was showing that we have already started genomic assisted breeding. Also genome-wide association working to study the genomics, means to study the mechanism of these all quality traits. So this is a rare opportunity. I used to say, you all collaborators, you phenotype as per your assigned trait in few centers, then you associate with the headquarters, so that we will be able to map the genes which are already not mapped, so that then only we can use in the genomic assisted breeding program. Similarly also I can say that for improvement, simultaneous improvement of the traits, you have to combine different centers, different traits, and design your best population, your training population, so that finally among the different center, ultimately within using somewhere in the speed breeding facility, now we are developing the speed breeding facility, where in all the center, whatever they are combining multiple traits, ultimately in the advancing generation, we will be able to intermediate the promising elite genotypes, so that the cycle can be advanced there, so that a good planning will be around the center will be done, as we are discussing in our key group meetings. So that will be taking place, and you have to take the advantage of that, and we can achieve, definitely I hope we can achieve the success in the coming few years. So this is a challenge, unless if you will be moving in the same pace, we cannot achieve target. Only we will be coming up with 22 million tons by 2047. As you know, the declining area is a very concern. So it is the main thing is that you have to see the profitability. Unless it is profitable to the farmer, they will not be believing your technology. So therefore, we have to improve, we have to add value to the variety. Then only they will be able to cultivate. And another very important is that the processing of this product, the farmer they cannot take to their plate. The cost is is very high. It's trading of this product is through like online everything. The cost is, once it is considered as a poor man's diet, it's now considered as a rich man's diet. So it is to be computed, it is to be changed. It's the way it is so high cost, the common people they cannot get. So that is you the people where you can make it profitable. If the yield will be very high, you will not be able to, farmer will not face difficulty in competing with other crops. So the genotype should be like that. It should be multiple biotic abiotic states, and biotic one means this biotic one, then will be actually worthy. And the staple crops like rice, wheat, maize, the millets are rich. If we can supply this supplement in this food plate, it will be a great for our citizens, all the citizens, if it will be available. So I hope by the end of the 2047, what we are thinking will be realized, and we will be achieving 35 million tons target. Though maybe the area may be stagnant, maybe little bit more, but our target will be achieved, and that is my view. And you take the advantage, we collaborate, you properly collaborate with the headquarters. Now they are having, we are strengthening that institute, all the latest phenotyping facility, genotyping facility will be enriched very soon, and you are having manpower, so all both by collaborative approach we will be able to get our target within the targeted period. So this is the thinking from the government side, I want to say you. So thank you very much. I wish we are very fruitful and successful conduct of workshop. Already I forgot to say you sir one thing. Already for small this sorghum and small millet, 3 days continuously we have interacted, and we have done the pre-group meetings, as well as for per millet also 2 days. So thoroughly we have already interacted each other, and we have discussed the key points. So I think this part will be very clear to you all. So thank you very much. Thank you.
Thank you, sir, for your enlightening words. Now, next on the program is the release of publications. We request all the dignitaries on the dais to join for the release of publication. The first publication is from ICAR IMR, book entitled Importance of Millets and New Technologies, honored by Drs. R.R. Chake, N. Kannababu, R. Venkateshwarlu, and C. Tara Satyavati. The second one is latest millet production and processing technologies, honored by Drs. R.R. Chake, C. Tara Satyavati, I.K. Das, G. Shyam Prasad, K. Hari Prasanna, Raghunar Singore, and N. Spandita. Third one is an extension folder, a handout on farmer producer organization. This is in Hindi, compiled by Dr. Sangapa, Mahesh Kumar, B. Rafi, Abhi Singh, K. Srinivas Babu, and C. Tara Satyavati. Again, another extension folder, a handout on farmer producer organizations in Telugu, compiled by Dr. Sangapa, G. Meghna, B. Rafi, K. Chandani, E. Karishma, K. Ramakiran, K. Srinivas Babu, and C. Tara Satyavati. And one more extension folder in Telugu, which is a profile of farmer produced organizations promoted by ICRIMR in Andhra Pradesh, compiled by Dr. Sangapa, K. Chandani, D. Rafi, E. Karishma, K. Ramakiran, K. Srinivas Babu, and C. Tara Satyavati.
Thank you everyone. Now the publication center, ICIR Agri-Farm Pearl Millet Coordinating Center, newsletter under the name Pearl Millet News, published by ICAR Agri-Farm Pearl Millet, volume number 13, 2024, edited by Dr. C. Tara Satyavati, Dr. Vikas Kandelwal, and Dr. Supriya Amrawat. The second one is the booklet named by Summary of Research Experiments 2023-24, compiled by Dr. C. Tara Satyavati and others.
Next publication is a book from ARS Anandapuram, ACRP on pearl millets, Andhra ARS Anandapuram, Research Accomplishments and Future Outlook, authored by Drs. L. Madhavilata, C.V. Chandramohan Reddy, R. Narasimhan, P. Shanti, B. Sahadeva Reddy, and I. Bhaskar Rao. And one more publication, Varieties and Technologies developed by ARS Perumalpalli in Finger Millet, authored by Drs. M. Srivalli Devi, L. Madhavilata, T. L. Hemlata, B. Sumati, D. Vajanta, K. R. Tagore, and R. P. Vasanti.
The other publication by Center ARS Vijayanagaram, the booklet named Transforming the Lives of Tribal Farmers Through Tribal Sub-Plan Program. Authors are Dr. T.S.S.K. Patro, Dr. N. Anuradha, Dr. Y. Sandhyarani, Dr. U. Triveni, Dr. S.K. Abdul Salam, Dr. M. Swati, Dr. V. Rajendra Kumar, Dr. M. Divya, Dr. C.H. Mukunda Rao, and Dr. P.V. Satyanarayana. And another booklet named Package of Practices of Millets in Telugu Medium, authored by Dr. T.S.S.K. Patro, Dr. N. Anuradha, Dr. V. Triveni, Dr. M. Swati, Dr. B. Rajendra Kumar, and Dr. M. Divya.
The next publication are pamphlets from AICRP on Sorghum India, varieties suitable for
Madam.
Next is the release of pamphlets from AICRP on Sorghum India, varieties suitable for sorghum cultivation, authored by Drs. P. Pulibai, S. Aisha Praveen, L. Madhavilata, M. J. Kiranmai, K. Arun Kumar, and M. Shivram Krishna. And also from AICRP Small Millets RARS India, Package of Practices for Foxtail Millet in Telugu by Drs. M. J. Kiranmai, K. Venkatramana, R. Narasimhan, and L. Madhavilata, and Diseases and Insect Pests and Their Management in Foxtail Millet in Telugu by K. Venkatramana, B. Chaitanya, R. Narasimhan, M. J. Kiranmai, L. Madhavilata, and M. Shivaramakrishna.
There are two booklets published by Agri-Pes in Jagdalpur and also a book. The first booklet named Mutation Breeding in Cover Millet, authors are Dr. Praful Kumar, Dr. P.S. Neetham and Dr. Narendra Kumar. The second booklet named Quality Seed Production in Small Millets, a database of Akrip on Small Millets Jagdalpur Center. Authors are Dr. Praful Kumar, Dr. Narendra Kumar and Dr. P.S. Neetham. The book named Research Profile Akrip Small Millets Jagdalpur. Authors are Dr. Praful Kumar, Dr. P.S. Neetham and Dr. Narendra Kumar.
Last but not the least, another release of publications is a publication from ICRIP on Small Millets Rural Agriculture Association, Kolhapur. Pamphlets on crop introduction, nutritional importance and improved production technology in finger, foxtail, little, barnyard, proso and kodo millet. Individually, Drs. Yogesh Ban, Dr. A.A. Pisal and others. And also a booklet on activities for awareness, production and area expansion of millets in Maharashtra, IOM 2023 and glimpses, authored by Dr. Yogesh Ban, Dr. A. Pisal and others. We thank all the dignitaries and also congratulations to all the authors of these publications. Thank you.
So, good morning. We have come to the next part, that is felicitation of the retiring scientists. We all know that having seniors with rich experience is a great privilege for all of us in guiding us in the right path, right? So it is our proud privilege for the Millet fraternity to facilitate such seniors who have done great works and retiring in this year. I call upon Dr. P.S. Shekhawat, Professor Agronomy in ICRIP Millet and the Director of Research of Swami Kesavanan, Rajasthan Agriculture University, Bikaner, to come to the stage to be felicitated by the dignitaries of the dais. Dr. P.S. Shekhawat, sir, please. He is the Professor of Agronomy in AACRP Millet and the Director of Research of SKRAU Bikaner. I request Dr. P.S. Shekhawat. We are felicitating Dr. P.S. Shekhawat with a kandua, a citation and a memento. Please join us in clapping to felicitate Dr. P.S. Shekhawat. Thank you very much, sir. Thank you all the dignitaries. Again I call upon Dr. Kailash D. Navagire, Sorghum pathologist, Sorghum Research Station, Vasantarao Naik, Marathwada Agriculture University, Parbani. In his immense contribution in the area of plant pathology and guiding many students for masters and doctoral programs. We have Dr. Kailash D. Navagire, Sorghum pathologist, in the stage.
Oh, hello.
We are happy to felicitate Dr. Kailash Navagire. And the next in line is Dr. Deepak D. Dudale, Sorghum agronomist, AACRP on Sorghum. I call upon Dr. Deepak D. Dudale, Swargam Agronomist, AICRP Swargam, Mahatma Phule Krishi Vidyapith, Rahuri. He is an agronomist and in charge for plant breeding in the center. AICRP Swargam, Mahatma Phule Krishi Vidyapith, Rahuri. Dr. Deepak Sundari. It's a proud privilege for the Millets fraternity to felicitate and honor Dr. R.K. Pandya, Principal Scientist and Head Plant Pathology, AICRP Pearl Millet, College of Agriculture, Rajmata, Vijay Raja Sindhya, Krishi Vishwa Vidyalaya, Gwalior, in his immense contribution in the area of plant pathology and guiding many students for masters and doctoral programs. Though Sir is not with us because of some medical emergencies for his wife, we pray God for her good health. And we call upon Dr. K.V. Raghavendra Rao, Chief Technical Officer in ICR, Indian Institute of Millet Research. It is our proud privilege for the Millet fraternity to felicitate and honor Dr. K.V. Raghavendra Rao, Chief Technical Officer at ICR Indian Institute of Millet Research, for his immense contribution in organizing group meetings of AECRPs and other activities in the coordinating cell since 1999. Dr. Raghavendra Rao was having immense contribution in organizing this such group meetings since 1999. Thank you, sir. And in this line, it is a proud privilege for the Millet fraternity to felicitate and honor Mr. O.V. Ramana, Chief Technical Officer at ICR Indian Institute of Millet Research, Hyderabad, for his immense contribution in statistical analysis of AICRP center data and many other activities in the coordinating cell since 1994. Though Sir is not with us, we remember him and honor him. Thank you all the dignitaries in the dais. Moving on, I request Dr. Savada Jayalakshmi Devi, Honorable Vice Chancellor of Andrew, to address this gathering as a distinguished guest. Vice Chancellor, Madam, please.
Good morning to all. It's my pleasant privilege to welcome the esteemed chief guest of today's function, Dr. Sharmaji, the Deputy Director General of ICR. And also I thank sir for selecting Andrew for this workshop, sir. And also I welcome all the dignitaries, and also I am thankful to all the ICR team for being with us in the Ajay N.V. Ranga Agricultural University. We all know millets have significant place as food and fodder crops. Though they are the traditional crops, they are now gaining importance because of their nutritional values. That's why they are called as nutri-cereals, wonder crops or miracle crops. And in recent years, the millets, because of their climate resilience and also tolerance to pests and diseases or biotic and abiotic stresses, they are being grown in rice fallows, and also they are being encouraged for the value-added products and also feed and fodder for the animals. The initiatives taken by the central government and state government, they are supporting the farmers, supporting the startups, and also supporting the FPOs. That's why the farmers are also being encouraged to grow these millet crops. We know all these millet crops are the traditional crops, because if you recall our history, if you take Andhra Pradesh as such, each district is identified for one millet crop, because the rice is the major crop, or Andhra Pradesh is the rice crop because of the rivers and other irrigation systems. Though that is there, all these millets are identified as the important crops of Andhra Pradesh. Because if you take the important districts, the Kurnool district, the previous Kurnool district is known for sorghum and korra. And similarly, Anupur district is known for bajra. And Chittoor is known for ragi. And small millets is for Mandal district. And Vijayanagaram is known for ragi and other millets. Like that, these are the traditional crops. We used to have these traditional food habits as being the millet, one of the important staple food of our system. But in recent years, we have forgotten because of the rice played a major role. But after COVID, everybody is very cautious about the health and taking the nutritional food. Even the government also encouraging instead of food security, now they are emphasizing on the word nutritional security. So everybody is cautious about the nutritional security, nutritional health, or having the long health with the nutritional food. So like this, we are now going with the millets. As you have seen the presentations of both Tara Satyavati Madam and Mansudana sir, how best we can have the millets in our system. So millets are very important food. If you take millets in at least in one meal, we can encourage the farmer. Because being the scientists, we are supposed to encourage the farmer. When we take our meal, one of the meal at least in the three meals of the day, at least if the millets are encouraged as one meal, the farmer will definitely get the support price to the produce he is growing. So like this, we scientists, we need to encourage the farmer, because the government also giving initiatives and incentives by growing these millet crops, and also encouraging the farmer because they are introducing these millets in the public delivery systems. If you take this ragi, it is now being given in the public delivery system. Like this, if the government also encourages the farmer by keeping these millets in public delivery system, and also in the FPOs and the farmers and the women and all these, if they are being encouraged, definitely millet will be one of the healthy diet in everybody's life. And also, now we are encouraging the millets because a lot of research is going on at ICR level and also at Anglo and also at the other state universities and also at academic centers. As we scientists having the lot of encouraging research, we should be in a position to encourage the farmer how best we can have the more productivity and also more tolerant to different disease varieties, and also we have to encourage the farmer to have more value-added products which are millet-based value-added products. Unless we have the entrepreneurship from the millet side, or the farmers or the youth or even women also to be encouraged as entrepreneurs of millet-based value-added products. Because in the Kurnool district, one of the entrepreneur, the farmer, Ram Subba Reddy, he was credited and he was appreciated by our Honorable Prime Minister in the Mangi Bag. So like this, if the farmer or any entrepreneur having these millets as the one of the important crops to be grown or as the value-added product, definitely he will be gaining a support price and also lot of profit from the millet crops. So as scientists, we know very well all these state and universities, academic centers are doing a lot of research, and also I thank ICR for sanctioning 4 academic centers. So in our university we have 4 academic centers, one for sorghum, then small millets, ragi and bajra. All these scientists, they are doing a lot of research work, sir. Totally 19 scientists are there in our university doing on these millet crops, and totally since 1964, the establishment of the university, 76 varieties were released. The varieties which were released in our university are very well known at national level, sir. The important crop, sorghum, is previously affected by striga and charcoal rust. Striga was the major problem in sorghum. At that time, our variety N13, which is very important striga tolerant variety, even now also it is taken as a national check, sir. We are proud of our varieties N13. And also recently bio-45 varieties were released, that is Bajra ADB4, which is from the Anandapuram, and the bio-45 varieties like Indravati, which is also released by our Honorable Prime Minister of Narendra Modi ji. Like this, we have many varieties, Indravati, Padmavati, Vakilam, Gavada, Venadu. All these varieties are being released or they are being cultivated by the farmers from Andhra Pradesh. And also there are different varieties which are now being encouraged by the farmers. There is NTZ, Nanda and Telangana, 2 varieties, which is the sweet sorghum variety, because we have two types of varieties, yellow jawar and white jawar. Most of the food habit, they prefer the white jawar. Therefore, our Angara Kalyanana varieties are being preferred by the farmers and also by the human people, sir, because they prefer the white roti. So like this, all these millets are having a much importance. So we as scientists, we should encourage the farmer, and also we should have the food habit of having multi-grain or the millets in our meal, and also we should encourage all rural youth and farmers to have the nutritional diet by having the millet-based nutrition. And even our university also having a patent for the multi-grain cookies and also millet-based products, sir. Like this, our university is doing lot of research. I thank ICR for giving the opportunity to encourage our scientists for going with the millet research, and also whatever the support which is from the ICR side, we are highly thankful to sir, and also we request you to support the Academy and Agriculture University, which is going further for encouragement of the farmers and also for the students, sir. I thank the opportunity given to me by the organizers, and also I thank all the audience. Thank you very much.
Thank you, madam, for addressing the gathering. Now, it's time to listen to our chief guest. For this, we request DDG Crop Science ICR New Delhi, Dr. Millets Raj Sharma, to deliver his inaugural address.
Good morning to all. Honorable Vice Chancellor, Dr. R. Sarada Jayalakshmi Devi, my distinguished colleagues from Council, Dr. S.K. Pradhan, ADG, FFC, Dr. C. Tara Satyavati, Director, Indian Institute of Millet Research, Dr. Madhusudan, our coordinator of Millets, Dr. P.V. Satyanarayana, Director Research, Angarao, Dr. B. Somati, Associate Director of Research, Tirupati, Dr. M.B. Ramanna, Associate Dean of this particular campus. We have with us our distinguished directors from Indian Institute of Seed Science, MAU, Dr. Sanjay Kumar, Dr. Lakshmi Kant, Director Vikas Almora, distinguished members of Project Advisory and Monitoring Committee, deans and directors of the university, statutory officers, distinguished delegates from Indian institutions as well as from CG centers, staff, students, ladies and gentlemen, press and media. First of all, I would like to welcome you all for this particular combined meeting of AICRP on pearl millet and millets. Probably this is for the first time we are doing such combined meeting at this very beautiful campus of Angara. A lot has already been said about pearl millets and millets, and I would like to place on record my congratulations to the Director Dr. C. Tara Satyavati and also Dr. Madhusudan for their excellent presentation. I could see that during these past 4 years, the level of presentation has gone to a different standard. And what is expected from a presenter, that is being included in a short presentation of 10 to 15 minutes so that all of us can become aware of what is happening and what has been done during past 1 year. Coming to the most important crop among millets is the pearl millet, and we know that this particular crop was domesticated more than 4,500 years back. It's such an important crop that around 90 million peoples of the world, they survive on this particular crop, particularly in Africa and India. And it accounts for more than half of the total millet production. We should be knowing that, and I am sure that you all are aware, being millet scientists, that the systematic research on pearl millet was started in 1936 in the USA, which led to the development of improved lines and improved cultivars. And this is the crop in which most widely used cytoplasmic male steroid system known as A1 was used. If you see literature, now we have A2, A3, A4 and A5 systems of cytoplasmic male sterility, but those systems, rest of the systems are not stable. Therefore, most of the emphasis for hybrid development was given on using this particular male sterile system, which is A1. And that is why in most of the meetings and also during the presentation of director in ICAR, I told that if we innovate some other stable male sterile system in this particular crop, that would be a real miracle. And that should be the task of our future targets of this whole group, which are dealing with pearl millet. A lot has already been told by our coordinators and director about pearl millet and about its spread across 13 states, 18 coordinating centers and different programs. But the first hybrid, which is named as HV1 hybrid Bajra 1, which was developed in India in 1965. So you can imagine since how long we are working on this particular crop, and it is a crop of the choice for the breeders because of its significance in our agriculture. In spite of the fact that this crop is being domesticated for the past more than 5,000 years, we still have many problems. And major problems which I could see is that there are certain insects like white grub, shoot fly and stem borer, which are still number one problems, and our targets should be directed towards identification of sources of resistance through our CRIP centers, and those sources of resistance to insect and diseases like downy mildew, blast, leaf spot. And I would say that multiple sources of resistance to insect and diseases should be our target now to identify. Not only that, we should pass on these sources of resistance from entomologists and plant pathologists to the breeders so that they can use in their breeding program. But we cannot use these in our breeding program until unless we study the genetics of inheritance of these traits among different varieties or lines. Based on their genetics, one can design a proper breeding strategy, how we can introduce the genes or the QTLs present within these donor lines in the cultivated one. And now we know that breeding has become a very important and easy tool with the advent of availability of genomic resources in these crops. And pearl millet is a very good crop with 2 and 14 chromosome number, but genome size is around 2 GB. But we are fortunate that this genome is already decoded and sequence is available in public domain. And if you see in literature, more than 30,000 genes responsible for different traits have already been identified in this particular crop. So another important approach why this group can be to use computational method to identify the genes and do validation in different systems. And now validation can be done by different approaches. By you can transfer the genes in different cultivars or maybe in heterologous system like Arabidopsis, or you can knock down the genes by using CRISPR-Cas and genome editing approach. Since pearl millet, we are interested to develop hybrid which can surpass the yield of existing hybrids and varieties. The role of apomixes is very, very important. And we can use this particular approach to produce true breeding hybrids or CNS lines. And nowadays, using this particular genome editing approach, if you can edit SNH3 genes, which is responsible for pairing of chromosomes in a cell, can easily produce double haploid apomictic plants. So, not only that, this crop has become a modern crop for using marker-assisted selection and genome-wide association mapping, but also a very important crop for biofortification. And this is the very first crop in which we have developed the minimum standards for producing hybrids for zinc and iron biofortification. You know that these crops are such an important that we have celebrated International Year of Millet, and there is a specific announcement by Government of India in last year's budget to develop Global Center of Excellence for millets at Indian Institute of Millet Research. And for all these millets along with pearl millet, there is no dearth of money now, as we are being told by the Honorable Vice Chancellor that we have even 4 AICRP centers of millets are established in this particular university, which itself told us the significance of these crops for Andhra state and for this university. And we are committed to strengthen these states because of their excellent output in terms of development of varieties which can be grown at national levels. Similarly, we have another component of millets which Dr. Madhusudan is taking care of as a coordinator and which was started way back in 1969. So, these millets, we have seen their significance, and all these diseases and insects which I told, like production constraints of biotic and abiotic stresses like shoot fly, stem borer, grain mold and anthracnose. And another very important pest, striga, which is mentioned was made. They are the traits of interest to all of us, in spite of the fact that work is going on these particular traits for the past many years. Still we don't have solution. But now with the availability of the technology with us, we can go ahead with introduction of genes for resistance or management of these traits by other practices. And we have seen that these crops are also affected by abiotic stresses like heat as well as drought, cold, and very important trait like nutrient use efficiency. Very important crops can be grown in the short duration, but still I think post-harvest processing of these crops are very, very important, and for which we need to develop various new protocols so that we can develop farmers-friendly post-harvest techniques for their processing and utilization. Luckily, we have very good genomic resources available with us, and 2 months back I was at TNAU where all the germplasm are related to finger millets were grown at one center. Around 7,000 germplasm lines were planted at Tamil Nadu Agriculture University by NBPCR to phenotype all those lines and remove duplicates among the germplasm. You know that as a breeder or as a millet scientist, we need more and more germplasm lines, and that is available with us at National Bureau of Plant Genetic Resources. For a breeder, genetic variability is the most important traits which should be available in the crop which they are studying. So if you have genetic variability in terms of germplasms, which we have in case of millets, you are lucky. If you don't have, then you have to create by mutation breeding or by using other approaches. And hybridization and crossing is one of the most important and easiest methods of creating variability among species. But unfortunately, it's very difficult to do crossing and hybridization in millets because of its small flower size. Therefore, 4 years back, I told that there should be a special program at Millet Research Institute to study the floral biology of all these millets, and I could see that that is one of the area of research which was included by Dr. Madhusudan in his presentation. I will further go and advise to both the coordinators and directors, and also to all of you, and particularly the breeders, that develop the product profiles of all the millets which are specific to a particular region. And in product profile, you have to consider all the traits which we need in a particular variety specific for that particular region. So those exercises should be done during these discussions, wherein we can plan what type of crosses will be developed by X KICRP center or Y AICRP centers, and how and what mechanism we should adopt to share advanced breeding lines and pre-breeding lines which were shown by Dr. Madhusudan and Dr. Tara Satyavati, how we can distribute those pre-breeding lines and advanced breeding lines based on the availability of hotspots of different biotic and abiotic stresses across the locations, across different AICRP center. That's the beauty of AICRP programs among all the crops. These are the largest networks of crops existed in the country. And this network is very large. I could see that more than 220 scientists or participants have registered for this particular meeting. So, if all of you are coming under one roof for 2 days to discuss about the priorities of research in millet crops, then priorities should be fixed, and my request to the members of, expert members of Program Advisory and Monitoring Committee, that try to develop this program in such a way so that we can eliminate the problems which existed in these crops and are being faced by the farming community. The mentions were made that there is a very short shelf life of millets after we make their product. And during my discussion at Indian Institute of Millet Research, I told that you have to reorient your program and identify germplasm lines which are having low rancidity. To reduce rancidity, if we don't have many lines, then we can use genome edited approach. Genome editing can be done to edit lipoxygenase genes, LOX genes, and in most of these crops, around 2 to 3 genes or LOX genes are present. And if you edit these genes, you can develop lines without rancidity and low rancidity. But for editing of those genes, lot of work is required, and that work has to be done either by university through students or Indian Institute of Millet Research. So all these 9 millet crops which was listed here, we need to develop transformation protocols for using in genome editing. We should have a very well designed set of genes which has to be edited, and also the genome sequence of all those lines which need to be edited to identify the number of copies of genes present within the genome. As a biologist, you know that for each and individual traits there are many genes which are responsible for those. Many of these are present in gene families or gene copies on different chromosomes. And for editing, if you edit one gene, the another gene will become active. Therefore, you have to identify all the copies of genes within the genome and develop your approach in such a way, so that in one go you can edit or knock down all the genes together and get a phenotype or desired phenotype like free of rancidity. So, in these crops, not only genetics and plant breeding is important, but we were told that 20 to 30% is yield gap which we are facing and getting in these crops. For that, we should develop very good agronomical practices, and all agronomists and soil scientists should try to develop mechanisms and design an experiment to develop package of practices in such a way which can be communicated to the farming community to reduce that yield gap. So development of good agricultural practices is one of the most important targets now, in spite of the fact that the potential yield is very high in all these crops. We are unable to achieve that potential yield because because of yield caps and certain problems in these crops. So similarly, for our plant protection scientists, I would like to suggest that in spite of the fact that we have very good institutions involved in these crops, the virulence analysis of different races of many of the diseases as well as biotypes of different insects is not known. Why don't you concentrate to do some basic work on the genetic variability present in the pathogen, not only on the host, the pathogen or in the insects, and based on that identify your resistant lines and deploy those genetic material across locations based on the prevalence of different races or biotypes of insects in those regions. They should learn more from the major crops like rice and wheat, how the research is being carried out and being followed in the country. You know the gene deployment strategy of wheat, which is very well known in our country and is given as an international examples. That is done based on the identification of races prevalent in different locations by Indian Institute of Wheat and Barley Research, and based on race flora and their prevalence across the location, we identify genes and deploy those varieties which contains those genes to contain the onset of epidemic of rust. So we can learn from those methodologies of cereal crops like wheat and use those methods and strategies in millets, because millets are becoming now more and more important for our food and nutritional security, and that's why government is giving special emphasis on these crops, putting a lot of money on these crops because of their climate resilience in nature and also their availability across the country. So I would like to request this whole group that during your discussion give more emphasis to the basic concepts and basic science along with your applied aspects. Because applied aspects we have to develop package of practices and give to the universities. Through universities it will go to the farming community. But basic research on various aspects which I just now narrated will definitely give you solutions for the problems which are present in these crops. So my best wishes to all of you for these 2 days deliberations, and I am sure that the recommendations which will emerge from this discussion will definitely lead us to reorient our future research program looking at the challenges ahead. So my best wishes to all of you, and thank you very much for attending this meeting.
Vice Chancellor of Acharya Engineer of Agriculture University, to kindly present the memento to Dr. T.R. Sharma Sir, Deputy Director of General CS, Indian Council of Agricultural Research, New Delhi. Give a big round of applause. Also, I request the ma'am herself to kindly present these tokens to Dr. S.K. Pradhan Sir, Assistant Director of General FFC, ICAR, New Delhi. Now I humbly request Dr. C. Tara Satyavati Ma'am, Director ICAR, IIMR and Project Coordinator for Millet to felicitate Dr. R. Shabha Jalakshmi Reddy Ma'am, Vice Chancellor of IIM Group. Let's heartily applaud them.
Madam. Yeah. Sir. Madam.
I also request the Tara Ma'am herself to felicitate Dr. P.V. Satyanarayana sir, Director of Research and Group.
ఇవన్నీ నేను నిలబెడుతూ వస్తాను ఫోటోలో తిరగాల ఆ పక్కకి.
Joke వచ్చేసి. బాబు. Joke వచ్చేసి. బాబు. Madam.
Now I would like to call upon Dr. P.V. Satyanarayana sir, Director of Research and Group to kindly present these mementos to Dr. C. Tara Satyavati Ma'am, Director ICAR, IIMR and Project Coordinator for Millet.
హరి హరి కొంచెం సైడ్ దిగు అవదరా మచ్చా.
Sir, here.
sir.
Now it's my privilege to request Dr. R. Madhusudana sir, Project Coordinator, Sorghum and Millets, to facilitate Dr. M.V. Ramana sir, Associate Dean, Sri Venkateshwara Agricultural College, Tirupati.
Sir.
Now I request Dr. V. Subhuti Ma'am, AGR RARS Tirupati, to kindly present this token of gratitude to Dr. R. Madhusudana sir, Project Coordinator, Sorghum and Millets. Please cheer them up with your hearty applause. I also request Dr. P.V. Satyanarayana sir, Director of Research and Group, to kindly felicitate the token of gratitude to V. Subhuti Ma'am. It's a moment where I request Dr. C. Tara Satyavati Ma'am to felicitate the token of gratitude to Dr. V. Subhuti Ma'am, AGR Tirupati. As we close this event, let's heartily applaud our honored guests, dignitaries, each and every individual behind this program, and thank you everyone for joining us in showing our appreciation and gratitude. Now I request Dr. J. Stanley sir, Principal Scientist, IIMR, to provide the vote of thanks.
Friends, we have come to the end of this inaugural session of this combined meeting on millets and I am here to express my gratitude and thanks to you all. On behalf of the organizers, AICRPs of Core Millet, Sorghum and Millets, ICR Indian Institute of Millet Research and Agriculture Research Station, Tirupati, of Acharya N.G. Ranga Agricultural University and all the millet fraternity. First of all, I profusely thank the Honorable Vice Chancellor Madam, Dr. Sarada Jayalakshmi Devi. Madam, thank you very much for giving her consent to organize this combined meeting on millets at this university itself, and also for her distinguished guests in this inaugural session and given an inspiring inaugural speech. Thank you very much, Madam. I profusely thank Dr. Tilak Raj Sharma, the Deputy Director General responsible for crop sciences in Indian Council of Agriculture Research. on behalf of all the organizers for his very presence as the chief guest of this program. Thank you very much, dear sir, for your inspiring speech and the guidance. All the Millet fraternity gratefully thank the Assistant Director General, Dr. Food and Fire Crops, Dr. S.K. Pradhan, for his patient listening to all the sector-wise presentations before this particular meeting and guiding us. We thank you for your gracious presence as a chief guest of honor and also for your address, sir. Thank you very much. I thank the Director, ICR Indian Institute of Millet Research and the Project Coordinator, Millets, Dr. C. Tara Satyavati Madam, who has taken all pains in guiding us and being with us all through this journey to make this combined group meeting a great success. Thank you, Madam. I thank the Project Coordinator of AICRP on Sorghum and Millets, Dr. Madhusudana, for doing all the things to arrange this function and for being the dignitary in the dais. I congratulate you, sir, for being the project coordinator Sorghum and Millets, as this is your first AGM after occupying this post. On behalf of all the Millet fraternity, we thank you very much, sir, for being with us. Though you were with us in the Sorghum for a long time, you have occupied this post and this is the first AGM you are occupying as a PC of Sorghum and Millets. Thank you, sir. We thank Dr. P.V. Satyanarayana, Director Research, Angro, a well-known scientist, as we all know, and an able administrator for all his support and for being among us and giving us these remarks, sir. Thank you very much, sir. And special thanks are due to Dr. V. Sumati, ADR, RARS Tirupati, and also for Dr. M. V. Ramana, Associate Dean, SVAC Tirupati, and all the people who have worked with them in various committees and meetings. And I hope these two people, this V. Sumati Madam and M. V. Ramana sir, they are very lucky to have a wonderful team with you, working in all the different aspects and doing all these arrangements so nicely. Thank you very much, Madam. Thank you very much, sir. And we thank the SV Veterinary University for granting this auditorium for this function too. On behalf of all the organizers, I thank the team RARS Tirupati and team ICR, IAMR and all the ACRIPs for all the arrangements made, be it the transport, food committee, or accommodation, the stage arrangement, the technical committee, and most importantly the finance committee, and also the Darshan committee. Special thanks for the steering committee and all the members. I thankfully acknowledge the presence of the directors of ICR institutes, especially Dr. Lakshmikant, who is the director of VPKS Almora, and the director Dr. Sanjay Kumar, the director of Indian Institute of Seed Science Mount, for your presence, sir, among us, and all the PMSC members, the VAC members, and many other dignitaries in this auditorium. Thank you very much, sir. We especially thank and acknowledge all the sponsors of this meeting. The silver sponsors are Cortiva Agri Sciences Seeds Private Limited and the Foragen Seeds Private Limited. The bronze sponsors, Acharya Injiranga University itself. They have to be called as diamond sponsors. More than that money, they have given everything for us to conduct this meeting. And also the bronze sponsors, the Classic Hybrid Seeds Private Limited, Crystal Crop Protection Limited, and the BASF India Limited. And the other sponsors, Coromandel International Limited. Dear sponsors, without your general support, we would have not done this meeting in this great way. Special thanks for all the volunteers, students who have worked in various capacities, especially for making the millet art in front of us and making this program very successful. The team RARS Tirupati needs a very good recognition for this. Above all, I thank all the scientists and professors who have come and graced this meeting from the length and breadth of this country. Thanks to all your organizational heads for allowing me also to attend this function. Without your valued presence of all of you, this meeting would have not got its glory. thank you very much of all the delegates. Thank you one and all. Jai Hind. Okay. I thank Dr. C. Ramana sir for his guidance, presence and all the arrangements made. Thank you very much, sir.
Thank you, Stanley sir. Now, we request you all to raise for our national anthem.
जन गण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड़ उत्कल गंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगल दायक जय हे भारत भाग्य विधाता जय हे जय हे जय जय
Thank you all. We have now come to an end of the inaugural session. The rapporteurs for this session were Drs. N.G. Vallarati, Dharba, K.M. Ganapati, IAMR, and P. Sanjana Reddy, IAMR. Thank you. We request you all to join for the Aging 2024 group photo, which is arranged outside this auditorium, and further the high tea. The technical sessions shall resume sharp at 12:30, and we request to be back at that time. Thank you.
Sir, no marks can lack the assessment, right? Good for the planners. At the screen, yes.
అందరివి వాళ్ళ డ్రైవర్లకి
మీకు ఎన్ని కావాలమ్మా? 2 కావాలా మీకు?
మేడం ఇదంతా లైవ్లో ఉంటుంది. ఆఫ్ చేయడంతో గంటలో లైవ్ ఇది రాదు, కెమెరా అటు వరకు రాదు.
లైవ్ వస్తుంది, లైవ్ వస్తుంది. ఏ ట్రైనింగ్ అంటే ఇప్పుడు తీసుకున్నాం మళ్ళీ ఆర్డర్ ఇప్పుడు ఆపాయాలి, ఇంకొక ఆపాయాలా గ్రూప్ ఫోటో వస్తుంది. గ్రూప్ ఫోటో अरे राघवेंद्र सर वो ले लो, प्रेजेंटेशन कौन लेगा? మార్నింగ్ ఒక మీడియం చెప్పండి సార్, కొంచెం కాపీ చేసుకోండి. మేడం థ్యాంక్ యూ. प्रेजेंटेशन होता है
Hema, dia sir kokkundi illa. Yes.
ನಂದು
ಕುಂಡಿ ಕೂಡ ಕುಂಡಿ తీసుకుని
ನಂದೇ ಅಡಿಬಿನಲ್ಲ ఫ్యాన్ అన్నీ ఇది అంటే లోపల అన్నీ అది పాలు పొద్దు నుంచి లేవు
టేస్ట్ ఉంటుంది అసలు డెవలప్ అయ్యి ఎక్కువసేపు వేస్తే పోతాయి. ఈ ఒక్కటి చెప్పాడు ఆయన అబ్బాయి ఫస్ట్ చెప్పాడు. ఏమో ఇస్తున్నావా పిక్చర్స్? Yes, sir. Yes, we can.
Just 1 feet, at least 1 feet.
కొంచెం చేయొచ్చు.
At least this is okay. 1 feet movement. Yeah, now it is a little bit, yeah, now it is visible. Now it is visible, now it is okay. No, I have this one, I turned this one. A second, now it is visible. Yeah, now it is clearly visible. Yeah, it stands here, na? Okay, sir. Okay, fine.
मैं आ रहा हूँ.
ఇస్తామండి. Sir, that tag we have missed, sir, that yes, sir. Yes, sir. I want to contact you also.
afternoon all of you. We are going to start the technical sessions. The first one is crop improvement. So I request all the delegates to come and occupy their seats. I request, it's a very important session, so I request all the delegates to come and occupy their seats. I welcome Dr. S.K. Pradhan, ADG FFC, ICR New Delhi, to be the co-chair of this session. This session is on crop improvement. There are 8 important presentations in this session. The first one being the plant breeding in pearl millet, will be presented by Dr. Vikas Kandelwal. Plant breeding Kharif and Rabi sorghum by Dr. C. Aruna. Plant breeding in forage sorghum by Dr. Venkatesh Bhatt. I welcome Dr. T. R. Sharma, the Deputy Director General, Crop Sciences, ICAR, New Delhi, to chair this session. This session is on crop improvement. We will have 8 presentations in this session, sir. The first one will be presented by Dr. Vikas Kandelwal, plant breeding of pearl millet, plant breeding Kharif and Rabi sorghum by Dr. C. Aruna, plant breeding forage sorghum by Venkatesh Bhatt, plant breeding sweet and high biomass sorghum by Dr. A.B. Umakant, plant breeding small millet by K. Hari Prasanna, Millets genetic resources management at LPPGR by Dr. Badal Singh, Millet genetic resources and management at ICR IAMR by Karna Venkatesh. I request all the presenters to note their presentations and come to the stage. I request all those presenters, namely Dr. Vikas Kandelwal, Dr. C. Aruna, Dr. Venkatesh Bhatt, Dr. A.V. Umakant, Dr. K. Hari Prasanna, Dr. Badal Singh, Dr. Karna Venkatesh to come to the stage. As I announced earlier, it's a very important session. I request all those delegates who are standing outside to come and please occupy the seats. Yeah, I request all the presenters to finish that presentation in 10 minutes. As we are already delayed in timing, I kindly request all the presenters to finish their presentations in 10 minutes, followed by 5 minutes of discussion. I hand over the mic to the chair for further proceedings.
Thank you very much. Along with my co-chair, Dr. S.K. Pradhan, I welcome you all for this crop improvement session in which various presentations will be made on various aspects of plant breeding. Since we have many presentations and time is less, so continue. I would request all the presenters to conclude their presentation 8 to 10 minutes, and all the participants will note down the suggestions, and we can have a discussion session at the end of all the presentation, and then we can discuss all those questions and clarifications at the end. Before we start, I request co-chair to say a few words and conduct the proceeding of this session. Dr. Pradhan.
Thank you, sir. Very good afternoon to all. Welcome to this important session. So before starting, I want to refresh you again. We had a thorough discussion about these things. May 27, 28 for millets, then for sorghum and small millets, we had thorough presentations on 6th of June, 7th of June and 8th June. In that we have reviewed the work plan, then this year work plan, then our center-wise reviews, and the result presentations we have already presented. The PIs they have already presented. You have also raised your questions there in the meeting. If anybody is having any questions also, you please note and ask those clarification again here. But I think all one-to-one we have discussed. So please note down your question, and we will be proceeding very quickly, allowing 6-7 minutes will be okay for each presenter, as we have thoroughly already discussed the matter. So thank you, I welcome you all. So let us invite the presenters. Dr. Vikas, I think will be presenting on 1 minute.
Thank you very much, sir. I announce Dr. P. Sanjana Reddy, Dr. Y.G. Ban of Kolhapur, and Dr. G. Girish from Hageri to be the rapporteurs of this session. Again, Dr. P. Sanjana Reddy, Dr. Y.G. Ban, and Dr. G. Girish to be the rapporteurs. Dr. Vikas, please.
Namaskar to all. Honorable ADG Sir, Dr. T.R. Sharma ji, Honorable ADG Sir, Dr. S.K. Pradhan Sir, Director IMR and Project Coordinator Sri Tara Satyavati Madam, respected Director ISSS Mau, Dr. Sanjay ji, Dr. Lakshmikant ji from respected. Okay, sir. So I am going to present the results for Kharif and Summer for Millet Coordinating Trial organized by the Project Coordinating Unit. So there are trials: IST early maturing hybrids for A1 zone, IST medium common trial conducted across A and B zones, IST late common trial conducted across A and B zones, AST medium conducted separately for B zones, AST late conducted separately for A and B zones, population trial conducted separately for A and B zones with common initial and different advanced entries, AHPT early maturing hybrids and population for zone A1. RHVT conducted separately for A1, A and B zones, SHT summer hybrid trial hybrids for summer cultivation with initial and advanced entries conducted in Rajasthan, Gujarat, MP, Maharashtra and Tamil Nadu for summer ecology. So this slide shows the trials number. For zone A1, 29 trials allotted. Out of these, 27 conducted successfully, 93% success percentage is there. In zone A, 73 trials allotted, and out of these, 68 trials conducted successfully, 93% success is there. And zone B, 69 trials conducted, out of these 63 conducted successfully, so 91% success is there. So total 170 trials allotted, out of these 158 trials conducted successfully, so 92% success is there. So next, so in initial hybrid trial, early, IST early, zone A1 for early maturity group, 26 experimental hybrids along with 5 checks evaluated at 11 different locations in zone A1 for early maturity groups. So 9 hybrids promoted for the next year of testing. These are MH2744, MH2749, MH2748, MH2743, MH2746, MH2754, MH2758, MH2745. Next. In initial hybrid trial for medium maturity group in zone A, 23 experimental hybrids along with 4 checks evaluated at 19 locations, only 1 entry, MS2785, promoted for the second year of testing. Next. In initial hybrid trial medium for zone B, 23 experimental hybrids along with 4 checks evaluated at 11 locations and 6 entries promoted for the next year of testing. These are MS2773, MS2784, MS2775, MS2767, MS2777 and MS2785. Next please. In initial hybrid trial laid for Zone B, 21 experimental hybrids along with 4 checks related at 11 different locations. 3 entries MH2808, MH2798, MH2795 promoted for the next year of testing. Next. In advanced hybrid trial for Zone B, 2 entries experimental hybrids along with 4 checks related at 8 different locations in Zone B. And both of the entries promoted for the third year of testing. These are MS2682 and 2705. Next. In advanced hybrid trial laid for Zone A, 13 experimental hybrids along with 4 checks related at 12 different locations in Zone A for late maturity group, and 3 entries promoted for the second year of testing, MS2709, MS2712 and MS2717. 2 entries completed third year of testing, so we will discuss in VIC for these 2 hybrids. 3 experimental hybrids along with 4 checks related in advanced hybrid trial laid for Zone B, and 1 entry promoted for the third year of testing, that is MH2717. Next, population trial, 10 experimental OPVs along with 6 checks related at 9 different locations in population trial for Zone A. Only 1 entry, that is MP637, promoted for the next year of testing. In population trial B, 10 experimental OPVs along with 6 checks related at 10 different locations, and none of the experimental hybrid promoted for the next year of testing. In advanced hybrid and population trial, 4 experimental hybrids along with 5 checks related at 10 different locations in A1 zone for early maturity group, all 4 promoted for the next year of testing. Next. In RHBT, 24 experimental hybrids and 6 varieties related at 7 different locations in zone A1 and A. Among early hybrids, RHB 223 recorded maximum grain yield. Among medium maturity group, BB 1852 recorded maximum grain yield. Among late maturity group, KBH 108 recorded maximum grain yield. Among biofortified group, HHB 299 recorded maximum grain yield. Among population, PUSA composite 701 recorded maximum grain yield as compared to other populations. In Zone B, 9 experimental hybrids along with 5 varieties related at 9 different locations. Hybrid 86M01 and MBH 4903 observed superior overall other hybrids in medium and late maturity group. Among populations, ABV 04 recorded maximum grain yield. Next. In summer hybrid trial, 13 experimental hybrids along with 4 checks related at 16 locations for summer ecology, and 3 entries MSH 405, MSH 406 and MSH 407 promoted for the next year of entry. Next. So I will go to discuss the plan of work for 24-25 next. Okay. So 9 entries promoted for the second year of testing in early maturing group.
Given because 2 minutes more.
Okay. And 4 entries for third year of testing. In late maturity group, 6 entries promoted for the second year of testing and 3 entries for the second year of testing. In medium maturity group for Zone A, only 1 entry to MH2785 promoted for the second year of testing. In population trial for Zone A, MP637 promoted for the second year of testing. In B zone, in medium maturity group, 6 entries promoted for the second year of testing, and 2 entries 2682 and 2705 promoted for the third year of testing. In B zone, in late maturity group, 3 entries promoted for the second year of testing and 1 entry promoted for the third year of testing. In summer, 3 entries promoted for the third year of testing. Next, please next. So 2 entries completed third year of testing. These are MS2626 and MS2631, and both recorded 10.11 and 9.63 higher grain yield as compared to the best check KBH 108, and it is also having a good iron and zinc content. Next. Both entries resistant against all the diseases and insect pests. Next. Thank you. This is all about Kharif 23 and summer 23.
Thank you. Interesting presentation. Very good. Next, madam.
Good afternoon, everyone. Respected chair, co-chair, and all the dignitaries. I will be presenting the progress under Kharif, sorghum breeding, which involves both Kharif and Rabi. Dr. Mathudana was taking care of Rabi earlier, and after attaining his position as PC, I have taken it up. Next, please. So these are the action taken and the major recommendations last year, sir. There was a recommendation to combine speed breeding with conventional hybridization to speed up the process. Yes, the efforts are initiated and one of our scientists is also trained in that and she has started working on that. And the second point is disease-resistant genes can be stacked and fermented in few elite lines. Yes, sir, we have used different resistant sources and we are combining the genes, resistant genes into elite background. And the new sources of resistance that have been identified in plant protection trials should be used. Yeah, the stable resistant sources which are proved over years that are being used in the breeding program. Now, next please. And coming to significant achievements, last year we could release one hybrid, say such 48, one dual purpose variety, one red sorghum and one yellow sorghum varieties. They were notified during last year, 23-24. Next please. And then the trials on the Kharif and Rabi, these are the trials which will be discussed further. Next please. And then before we go into the MLT trials, I would like to say, in brief, what we are taking up on the trait-specific breeding and also sharing of material and inter-institutional breeding programs. The first one is inter-institutional hybrid evaluation, where we have pulled the MS and R lines from different centers and made the hybrids which were tested, and the hybrids which are put in highlighted like in the red color, they were found to be promising and the seed of that is produced, and these hybrids are entered into IHT now as a combined product at the centers. Next please. And another activity is this is a interspecific derivatives, which one of our colleagues, Dr. Visarda, was working on that. She developed some of the restorers. These restorers we have distributed across centers so that they can use them in that R-line program. Actually, we tested them in the hybrid combinations. 29 hybrids were made, but the main purpose is to share the material so that they can use it to diversify the genetic base. Next, please. And another cold breeding activities, we shared the F2 materials from different centers. These are the trait specific like SFE, shoot fly, grain mold, and we distributed that F2s across the centers, and the selections on the right side they were made. So through shuttle breeding, next please, we could come up with some of the lines, like now they are in F6. In the last Kharif, we evaluated the F6 at 4 locations. And the last lines given in the last column, they were all promising. And now we will be combining, they are very stable across the locations and over the generations also. So these will be used further. And another set of F2s, again for P-line improvement and R-line improvement, these were shared during Kharif 23, and these were the selections made. Next please. And coming to trait-specific breeding, in Kharif, grain mold, shoot fly, downy mildew, and the development of colored sorghum. These are the important issues what we are targeting using at different centers. These are the material, the elite lines and also donor parents that have been used. Next please. And similarly in Rabi also, shoot fly, charcoal rot, rust, downy mildew, terminal drought, cold, aphid, shoot vine, stem borer. These are the important issues that have to be targeted, and these centers are working on each of the traits. And during 24 or 25, this advancement of this process have been taken up. Next please. So this is the trait-specific breeding activity under Rabi sorghum, which has been taken up by the centers which are listed at the down. And these are the different traits we were addressing, including the lines which are listed here. Next please. And other inter-institutional activities, that Malgandi CMS, the fertility restorers for alternate cytoplasm, that is really a big issue in Rabi sorghum. So we could identify 5 stable restorers, the germplasm and other lines. So these restorers were distributed across the centers, and we are now making a process with the elite lines to develop restorers for the alternate cytoplasm. Similarly, Bog Malgandi cytoplasm development is being taken up. Backcrossing is under progress. Then introgression of the staggering QTLs, 2 staggering QTLs have been identified as stable, and these are used in the used to generate the material. Like these elite lines, M35, SPP2217, these were used to integrate these QTL into that elite lines. And then exploitation of land races we are taking up, and then the evaluation of segregating material, which is trait specific, that is one we are addressing. Next please. Besides that, and ERA trials also we have initiated last year, which will go for 2 more years, and then inter-institutional hybrid trials for Rabi specifically, because Rabi hybrid development in Rabi is a bit lagging. We are concentrating more on that. So besides that, we are also taking up reel development from multi-parent process. Now 3 reels are available, and marker-resisted backcross breeding is also going on to incorporate resistances for important diseases which are listed here. The donors are listed. That is being taken up. Next please. And then MLTs, these are the MLTs that are under Kharif and Rabi. The list is given along with the locations and the number of locations that has used in the Indian meal. Next. And this is the range of grain yield and stover yield we could record in each of the trials. Next please. And when we see the performance in the AVHD, Advanced Variety and Hybrid Trial of Kharif, SPH 2021 one hybrid has done well in all the 3 zones. With 10% improvement in zone 2 and 5% in zone 1. Another hybrid SPH 2024 is also doing well in zone 2. Next please. And based on, we have a set of criteria which we have already discussed in the technical program. These are the genotypes that have been promoted in different trials. Next please. Again in the initial hybrid trial and initial varietal trial also, I have mentioned the genotypes that have been promoted based on a criteria. And the fifth, there is one IA SSVT, that is specialty sorghum trial, where we are looking for, besides yield, we are looking at any specific trait like high iron and zinc or color sorghum, also one specific trait what we are looking for. Next please. So there is one hybrid SPH 1974 and one variety SPV 2871. These two have completed 3 years of testing under AACRP, and these are the proposals are being submitted for this. Next please. And this is the specialty sorghum trial which I was mentioning, where we could find some of the lines with high iron and zinc. So they are in the 2nd and 3rd year of testings now. So this will be further tested, and promising ones will be identified. Next please. And coming to Rabi also we have the advanced and VHD, the promotions that are made under Rabi trials based on the criteria which are listed at the down. So these were identified, these were all discussed with all the centers also during the technical program formulation. Next please. And during Rabi, I just want to mention one thing, that there is one hybrid, Rabi hybrid SPH 2036. This is based on an alternate cytoplasm, sir. It's not Milo, non-Milo, A2 based, Malgandi based. So that hybrid is doing well over the existing hybrid, that is CSH 39R. So for 2 years it has shown 8% improvement. This is in the 3rd year of testing now. So hopefully we will get a good Rabi hybrid next year. And even in the initial trial, as you see here in the IHD, there are 3 hybrids which were tested, and all 3 were doing much better than the check. Next please. And in Rabi specialties sorghum, we have one Huruda trial.
Madam, one.
Yeah. Huruda trial. Huruda is like a special type of condiment which is used, delicacy which is used as delicacy. We have a trial for Huruda, pop and flake sorghums, colored sorghums, and high iron and zinc. Next please. So in Rabi, actually 6 entries have completed 3 years of testing, but we could identify 2-3 that were good across 3 years. Next please. And this is the technical program for 24-25. So we have regular MLT trials with these many entries, checks and locations. And then besides that, we have inter-institutional hybrid trial and evaluation of F4, which I was mentioning earlier, for shoot fly, grain mold and grain yield. Next please. And some new initiative which Dr. Masudana has presented in here, we are going to have one summer sorghum trial and also rice fallow, which is picking up area. Next please. So in Rabi also we have the multi-location trials, AVHD, IVHD, then specialty sorghum trials 2 and the parental trial, and one inter-institutional hybrid trial also we are going to take up, and once we collect the hybrids from different centers, we will be formulating that trial. Next please. And these are the publications we have total, it is across all the breeding group, so more than 6 or 15. Next please. Thank you sir. Thank you so much. Sorry, I had to
Thank you. Within due time we have completed. Next,
So in forage sorghum we have 2 commodity profiles. One is single cut forage, which is grown as rainfed kharif, and multi-cut forage, which is grown under irrigation. So it is cultivated in April. So next one please. So we have 2 zones. One is North India and the other one is South India. Actually it's rest of India, but we don't have testing centers in Northeast and Jammu and Kashmir. So Right now it is only South India. Next one. So we have 9 centers in each of the zones and we have 4 multiple location trials. Next one. So our selection criteria has been discussed in earlier programs also. Next one. So we have benchmark hybrids and varieties in both commodities of single cut as well as multi cut identified. Yes, next one. These are our national checks, and the details of the centers and the states under which they are coming are covered here. So next one. So we have here basically this is to say that we have more of hybrids in case of multi cut and more of varietal entries in case of single cut. So we have again more of private hybrids in multi-cut and public varieties in single cut. Okay, so next one, yeah, this is about the promotions which has been discussed. So next one, so yes, I think that's all. Most of the technical programs have been discussed. Okay, next we come to the pre-building programs. So in college program, the main important criteria is to have a high amount of production per day as well as the quality of the college. So our building programs are targeted improving these states and also multiple states such as stem board resistance, soil resistance, along with the yield and quality aspects. So we also look at lower lignin content using BMI lines and free sorghum to have more digestibility of the water and things so on. And in this regard, our centers who are all working on forage sorghum, they are making different process and they have advanced generations. I think we will not go into details. And then coming to specific problems, forage is a problem in especially in single cut forages. So our Hisar Center and Pantnagar Center, they are working on forage diseases. Next one please. So stem borer is a very important problem and Hisar Center is the hotspot for that. A very intensive program on making pedigree selection against this stem borer insects, and Pantnagar is also working in this area. Next one. And sulfur is important pest again in all kinds of sorghum, and Ludhiana, Patnagar and Udaipur, which are our major foliage centers, they are again using the different sources of resistance to fruit fly, some of the improved ones as well as the conventional ones, and trying to put in the background of the varieties which are adapted to their state. And the challenges which remain are basically that we don't have much of the genetic variability for the quality and yield, and also for multiple cuts in case of multi-cut sorghum. And another important thing is to develop hybrids. We don't have islands which have high yield potential. So now we are working on all these aspects in our program. Next one. Yeah, that's all. Thank you.
Thank you very much. Before time completion. So, please start.
Dignitaries on the dais and off the dais. I am going to present the progress and achievements in sweet sorghum, high biomass sorghum and brown red sorghum. Next. With respect to the achievements, this year we have released sweet sorghum variety CSP58SS. You can see the trait. I mean, what's important is it has a very good fresh stock yield or the millable care yield of which is ranging between 50 to 53 tons per hectare. And if you assume 50 liters per ton, this variety is going to give 2,000 to 2,500 liters per hectare. Next, another brown midrib sorghum variety CSP59 BMR. This has been notified for 8 states. And grain fodder yield ranging between 300 to 400 quintals per hectare, and dry fodder 12 to 12 tons. Next, so of the major recommendations during last AGM, the first one was to test 8 entries in advanced trials, and this has been complied with. And one more recommendation was to relook into the ethanol yields while formulating the product profiles. Sir, we have relooked into it and then we have changed it. And target for foliar diseases, especially during promotion of entries, it should not be more than 1, and this has been complied with, and we are checking the data, and then we are promoting only those entries which are less than 480. Next, these are the multi-location conducted on sweet sorghum, high biomass, and then brown red sorghum, apart from one exclusive trial on testing the single cut foliage yielding ability of the released private sweet sorghum hybrids being marketed as single cut foliage yielding hybrids in northern India. And with respect to the performance, you can see here SPV with a fresh stock yield of 49 tons against the check CSP 49 SS, the best check 42 tons. The superiority here was around 17%. And with respect to the juice yields, SPV 2890, which has recorded around 19,000 liters per hectare against 17,000 liters in the best check 49 SS, and superiority here was 12%. So SPV 3 was better for and also the juice yields. Next, and coming to the between 15.5% over the best check, 24 SS here, and then another value was also superior. And coming to the ethanol yields, 3 entries were found to be superior, and they have recorded close to 1800 and above liters per hectare, and 15% superiority over the best check 49 SS. Next. And on the hybrid front, the 2 hybrids were tested against the standard hybrid 20 SS, but these hybrids, they were superior, first stocking, and which was on par with the Czech hybrid. So nothing was promoted in this. And this variety SPV 2890 has completed 2 years of testing, and it has all the superiorities over the Czech, and proposal is being well put up. Next. And with respect to the high biomass sorghum trial, none of the test entities could surpass the levels of the best check, that is CSP 48, which has recorded around 31 tons of dry biomass per hectare. Next. And coming to the brown red sorghum trial, almost all the entries except SPV 3088 have recorded more than 10% superiority for both green and dry proteins. And with respect to the protein yield, also SPV 3087 and 3089, they have recorded 15% more protein percent than check top 3 BMR. Next, this is the result of the sweet sorghum genotypes which were tested against the private hybrid, which is the sweet sorghum hybrid being marketed as photo sorghum hybrid. And here we can see that CSV 47 of number 13, this basically is a sweet sorghum variety which has it has given 37, it was 37% superior compared to the Czech sorghum grains. See that here is the set. Next, this is trial which will be taken across 14 locations. That is national sugar. We have added from this location. Next, high biomass sorghum again. 8 locations in 6 states, and biomass sorghum again 8 locations covering 8 states. Next. Coming to the pre-breeding activities, IMR has shared 16 classes across 10 centers. Here the process for sweet sorghum covering high stock yield, bricks and insect pest tolerance apart from the brown character, and selections are being made, and they are continuously supplying entries to all the sorghum. Next, they have in their centralized presentations. Next. These are the overall conclusions from the ASAP trials. Next. Sir, coming to the trials and promotion of sweet sorghum for biofuels. So IMR has entered into a MOU, 5-year MOU with National Sugar Institute, Kanpur. So we have completed 3 years of testing. So they are testing the sweet sorghum in their premises, and also they are helping us in reaching to the sugar industry. Next. These are the results, 2-year results at MSI Kanpur. So the stocking of our genotypes range between 45 to 54 tons per hectare and the ethanol yield. And this practically they are doing at lab scale level, and the reported ethanols are quite remarkable, 2,000 to 2,500 liters per hectare. So practically they have done this. So next. So with their initiative, we could reach Balrampur Chini Mills, which is one of the biggest sugar factory in Uttar Pradesh, and they have tested sweet sorghum because they were not aware about sweet sorghum. So initially we just planted that in the return of sugarcane. There you can see that, and also you can see the height. I mean that is CSS 22 is our best bet available with the Indian sweet sorghum program. So the stock range between 60 to 70 tons per hectare in this sugar company. And the next one was Dalmia Sugars again in next. Dalmia Power Sugars. Here also they tested the sweet sorghum. Here you can see the director of National Sugar Institute along with the sugar mill staff, and both the mill management and also the farmers, they were quite amused with this crop. So this can go for biofuel and also as a very good forage. Next. Sir, IMR also is in collaboration with Pani Foundation being noted Bollywood actor Amir. So we have an MOU with them. They are sweet sorghum. They are promoting. You can see that our varieties in the farmers field, they have tested sweet sorghums in 7 locations in Maharashtra. Next. So this is the analysis. Our entries are performing very well in farmers fields, more than 40 liters per ton of ethanol. They have reported, this is the data, fermentation data from National Sugar Institute in Pune. And one of our entry CSV 49SS. So it gave more than 40 tons per hectare of stocking, and apart from that, it gave 3 tons per hectare of grains. So anticipated ethanol yields from this particular variety per hectare is going to be 3,000 if you club both the juice-based ethanol and also the grain-based ethanol. Next. So publication and recognitions. So I was invited as a panelist in the G20 Energy Transitions Ministerial, which was held at Goa in July 2023, and also I was felicitated by Pani Foundation recently for the biofuel promotion activities. These are the publications that I think. Next. This is the work plan, which is more or less continuous activities, and thank you. Thank you all.
Thank you, Dr. Umakant. In time, next presentation is Dr. Hariprasan. So you are given 8 minutes. Grace will be maybe 1 to 2 minutes.
I have 7 questions. Yeah, respected chairman,
respected chairman, co-chairman, dignitaries and delegates. I am here to present the progress of small millet breeding. Kindly excuse me for my voice because I have a severe infection. So these are the 7 crops or 7 jewels on which we are working, sir. Finger millet is having the maximum share of around 70 to 75% of the total area with around 12 lakh hectares. This is as per the third advance estimates of 2023-24 given by ADFW, and the remaining 6 crops they together constitute about 20 to 25% with about 4.82 lakh hectare and 4.34 lakh tons, and average yield levels have risen in the last 2 years from around 750 kg to 900 kg. Yeah, next please. And this is our the small millet network, sir. We have 13 centers in 11 states and 11 state agriculture universities catering to all the 7 small millets. And along with that, we have more than 20 voluntary centers. So out of these 13 centers, 9 centers started in 1986, way back along with the launch of the small millets at GKVK Bangalore. And some of the centers were recently added, like Mandya, Jagdalpur, Gulabpur, Bagay. Next, please. Yeah, this is the recommendations and action taken for the last AGM, sir. So we have only 2 sessions or recommendations. So one is that lines which did not get identified or notified may be utilized in the grading program as done. So this is a regular program, sir. The centers are already using these advanced lines in the crossing program. So some of the lines I have listed here which were tested in the last 5 years, and they were not promoted as varieties but they were used as parents and the crossing program. And the second recommendation was that potential test lines which are not promoted in the trials can be used in the basic studies like GWAS or Maximo and tagging agronomically important traits. So this also will be taken care in the future whenever we are constituting the macro population, when we are constituting the association learning panels. Next please. Coming to significant achievements, so we had 7 national releases last year, sir, 2 in finger millet, 2 in powder millet, and 1 each in forestal millet, barnyard millet, and little millet. Next please. And along with that, we have 7 state releases, sir, for different states, in 2 in finger millet and 1 each in forestal, cover, barnyard, little, and orzo millet, covering different states. Next please. Apart from this, we also have 6 finger millet stocks registered for as a trade specific genetic stock under NBPGR. Next please. Yeah, this is a trial session conducted and success in the last year, sir. So out of 7 crops we have 8 types of trials. Only in case of finger millet we have separate IBT and ABT. So altogether 124 trials were 118 trials were conducted with a complete of about 95%, and there was a high CV, so we could not use the data from around 35 centers. Next please. Yeah, this is a summary of all the trials taken up in the 7 crops. So total 10 trials including IVT and AVT. In case of finger millet we have separate zones. That's the zone 1, that is the southern part, and zone 2 is the northern part. So total 75 entries were tested under IVT, and under AVT 27 entries were there. And the total number of locations are listed here, sir, and the total states or the Indian territories covered are total 16. Next please. Yeah, coming to the results of individual crops, sir, so these were already discussed as ADG told, so I will not go into the details, so just the summary only I will be reading. So in case of IVT, as I already mentioned, 23 entries were evaluated in the 13 centers. So in the zone 1, none of the entries had superior performance compared to the best checks in FME 2, and in the zone 2, one of the entry FME 1254 and FME 1275 were recorded just a marginal grain yield and fodder advantage over the best check. Next please. In the advanced trial, in zone 1 we have no entries, sir. In zone 2, out of 5 entries evaluated in 8 centers, so one of the entry FME 1244, that is when we pick as Almora. So we recorded a grain yield advantage over the early check VL 376, and it is early also comparable to the VL 376. So this has been promoted, and over 3 years again 1 entry from Almora, that is FME 1209, it recorded a 5% grain yield advantage over the early check VL 376. So this has been put for the VIC. Next please. And the next crop, that is pastoral millet initial and advanced variety trial. So out of 98 entries evaluated in the 17 centers across 12 states. So 2 entries, FXV677 and FXV683, they recorded marginal grain yield advantage over the best check DHFT 109-3, and FXV680 was significantly early over the best check. Next please. And over 3 years, we had 2 entries, FXV647 and 652. FXV652 recorded more than 5% grain yield advantage over the best check. and it has been put for the VAC, and over 2 years, none of the entry was as per our criteria, so it was not promoted. Next please. We had 9 entries evaluated in 12 centers across 7 states. So 3 entries came in 581, 582 and 588. They recorded more than 10% of water advantage, but for graded there was not much significant improvement. Next please. And over 2 years we had 2 entries, 581 and 582. So both recorded more than 10% water advantage to reject serial 1. As well as and coming to multi-millet initial and advanced variety trial, so we have 16 entries were evaluated in the 14 centers across 9 states. So one of the entry will be 592 and it was early also. Next please. And earlier compared the checks, but we were not having performance, it was an entry with earliness, but there was a rural advantage. Next please. Coming to barnyard millet, here 11 entries were evaluated in 13 centers. So none of the entries had any growing advantage, but BMA 644, 645 and 646 and 648 that is more than 5%. We had 1 entry, BMA 638, but there is no growing advantage, so hence it was not. Next please. Coming to proposal millet, here 10 entries were evaluated across 10 centers in 8 states. So PMB 472 and 473 and 489 they recorded more than 5% growing advantage over the best check TNA 164 and 483 had more than 10% watering advantage also. Next please. And over 3 years 472 and 473 both had good growing advantage, so both have been put up for the VAC. And over 2 years 2 entries PMB 480 and 483 they were evaluated, whereas 483 had better watering advantage, so it has been promoted. Next please. Millet, so here 7 entries were evaluated in 5 states. So only 1 entry means BTV 31 and BTV 41, they recorded more than 10% watering advantage over the best check, but there is not much growing advantage. Next please. Over 3 years, we have 2 entries at BTV 30 and 31. So both were only marginally superior compared to the best check, but both had better watering. And there is a 3 years, sorry, and over 2 entries were there, but there was not much superiority for both greening and watering, hence they were not promoted. Next please. So this is a summary of the number of countries promoted from IVT to AVT. So from IVT to AVT, first year we promoted 20 entries and the 7 crops, and from AVT 1 to AVT 2 we are promoted 4. So total 24 entries will be evaluated the current season. Generation. So we have assigned to the different second column. So this was as approved by the DDG sir. So some of the training programs are like genetic improvement and figure. Then genetic improvement in proso millet, then blast resistance breeding in finger millet, and shooter resistance breeding in case of little pros and foxtail millets, and screening the small millet crops for the tolerance to banded millet. So these activities have been assigned to the different centers. So the breeding material generation and advancement of security and the selections are going on. Next please. So program taken up with the different centers in different crops, and not into the details. The traits also have assigned here as per the assigned breeding program. So more than 100 crosses have been this year in the different crops. Next please. Breeding nursery generation, advancement and selection.
Dr. Prasanna, you are using grace time.
Yes, sir, I will be completing 2 minutes. Yeah, and this is millet. Next please. Again, this is in case of proto millet, the mutation breeding material. Next, coming to the pre-breeding program, so this is the brainchild of our DDG sensor. So as 2 years back, we have assigned this connectivity to the different centers. So they are making the process as per the trace listed here. So the materials in various generation of F2, F1 as well as F2. So they are advancing the generations, and I hope within the next 2 years we will be having new material emerging out of this exercise. Next please. Next. Again next. Next please. Yeah, so this is a trade specific breeding lands identified in different crops in Srinagar. That is the Wildland Agriculture Research Station. Then 2 centers in the northeast have been added in case of finger millet, and similarly in other crops also we have added some new laboratory centers this year. Next please. And these are some of the new initiatives that we are going to take up one pre-IDP in case of finger millet. Next. Then irrigated or summer time we are going to take in case of finger millet and foxtail millet, which we are going to start from this year summer, sir. Then screening for high temperature in case of finger millet, which again we will be taking up in summer. And breeding materials, already we have collected material in finger millet and we will be sharing it with please. And these are the general guidelines. Please. Yeah, thank you, sir. Thank you for the extra time.
Thank you.
Thank you, sir.
Right, taking you used 13 minutes.
Thank you, sir. But I had 7 crores.
Next. These are the state-wise characterized 25,000 accession of sorghum against 5 checks. Next. These are the 25 agromorphological traits which we have characterized on which includes 9 quantitative and 16 qualitative traits. Next. These are the views of sorghum mega characterization experiment. Next. Next. These are the variability we have observed in the earhead shape and green color in case of sorghum. These are the glimpse of program of sorghum characterization. Next. With this previous year we also characterized entire NGB collection of barnyard millet and against the 4th check. Next. Based on 25 agromorphological traits which have include 10 quantitative and 13 qualitative traits. Next. These are the next. This is the list of total registered germplasm in millet. Next. These are the some germplasm which has been registered for some unique traits. Next. Next. Thank you.
Thank you.
So next. So around 49,000 accessions of millets we are conserving at AMR Gene Bank, sir. In that sorghum is the maximum one, that is around 27,000, and remaining I am presenting here, sir. Next. So during this year, so we multiplied around 5,800 accessions of different millets. In that sorghum was maximum, followed by foxtail. Next. So coming to the distribution, sir, as NBPGR has initiated charging of germplasm handling fee. So this year the distribution has been dropped significantly. So we distributed around 203 germplasm to our centers. So this is the output. So next. So as already presented by Dr. Badal Singh, so we characterized 25,000 accessions of sorghum at Akola. So next. And also, as DD sir mentioned, we are characterizing around 12,000 accessions of finger millet at TNA Coimbatore. So the process is still going on. So harvesting and post-harvest data is being collected. So these are some of the photographs of the activities conducted in TNA Coimbatore. Next. So under the DRP project, so along with Engineering Center and VPK Salmora, we are characterizing finger millet accessions around 2,500 accessions every year for blast diseases. So these are some of the photographs. Next. So, like for example, common entries resistant to blast diseases across all the centers have been identified, and these will be confirmed again in the next year, and then they will be shared with breeders for using the breeding program. Next. So this is the previous entries, promising entries from multi-location trials. So these are tested at 6 locations. For last, these again will be going for registration and sharing with breeders. Next. So same thing. Next. Technical programs. So again, most of the activities will be ongoing. So one thing is we will be going for exploration in the northeastern region, and then we will be augmenting some of the small millets from NBPGR. Next. And then there is one proposal where we will be going for characterization of all the 6 millets at Hyderabad. Next. Again, this CFP-AB program will continue at all the 3 locations. In this, 2,500 accessions will be screened, and then 2,500 accessions, one more trial will be there at Gudamalani Center, where we will be taking up screening of all the permanent accessions available at National Gene Bank. Next. So again, we will be multiplying some of the accessions at Hyderabad. Next. And there are some wide hybridization derivatives available with us, which were developed by Dr. Visharda. So we will be planting them for selection by breeders. Next. Again, the distribution activity will continue, and then conservation will continue. Thank you, sir.
So, participants, please, if you have any queries about promotion of the entries, 3 years based pricing entry, you have any query, we have a discussion. If still any question, please ask. I think so already discussed. Okay, please ask, please take the microphone. Please give the microphone.
Thank you. I'm coming from Intisat. I'm principal scientist to help with several brilliant artificial headquarters. I have not a question but a comment to what Dr. Aruna said about the difficulty finding molecular restaurants across cytoplasms, which is indeed a problem in terms of being able to diversify cytoplasms in sorghum in order to avoid any criticality, particularly in the case when the cytoplasm donor is attacked by or vulnerable to diseases and insects. So at research, we have started working on this issue, and we have identified several restaurants, more than 5, that were stable across cytoplasms and across environments. And my point here is we are able to share this material with IMA so that we can provide effort in terms of diversifying these cytoplasms and enhancing the accessibility. This is my point for the moment. Thank you.
Thank you very much. So, if you have, please.
Updating the breeding activities as well as specific breeding program and all the crops. One small suggestion is that a lot of material has been generated in these activities. They have a lot of breeding material. So these things should be tested for more distribution across
association, so that we can evaluate. That's one thing. And second point is that in small millets, the new seeds whatever that Hendrickson has told. Yes, somewhat difficult it is there. So I suggest you to go for other millets also. Foxtail millet and proso millet also simultaneously, at least from next year onwards, you can think of starting summer trials in all other small millets crop, particularly foxtail millet, little millet and proso millet. These three crops you can add it. So otherwise the program is very good. Thank you.
Thank you. Dr. Prasanna, please. So I have no more questions. So sir, please.
Okay, so very good presentations were made by all the speakers today, but while listening all these presentations, I noted down few points. And I would like to say that in case of Dr. Khandeval's presentation, very few entries were promoted to the subsequent years or next year. It clearly shows that there is not much attention given to the selection of parental lines for making crosses. So my suggestion would be that you need to select diverse parental lines for crossing in future and take the help of experts in this particular meeting so that you can create a lot of transgressive segregations which can be evaluated across locations and proceeded ahead for next term. In second presentation, sorghum speed breeding was mentioned. I would suggest that you have to standardize the speed breeding protocol and how many generations you can take it. That was not clear. In this case, you can take help of a preset also. And pre-breeding material which is being shared across locations should be evaluated for multiple traits in case of sorghum, and specific data to be presented next year during the annual workshop. Third presentation was by Dr. Bhatt on forage. It was very important and I would suggest that a strong collaboration with the IGFRI is required, particularly mentioning about multi-cut and stay green trait hybrid. Work on those and collaborate with the IGFRI for further evaluation on these particular lines. Sweet sorghum is very important now. It is becoming one of the most important energy crops and need to give more emphasis in coming years. In this particular crop, I will suggest that you need to have strong industrial collaboration. Otherwise, it will be very difficult to develop protocol for alcohol production. Plant breeding for small millets, although there are many crops, but looking at all the data presented by Dr. Prasanna, we have a little variation which exists in these crops, and there is a need to collect more number of genotypes from different parts of the country, develop core and mini core, and evaluate those core and mini cores across locations through AICRP Center. NBPGR presentation is good, I mentioned in the morning also, but they are concentrating only on one or two crops. In small millets, very little collection they do have, and I would suggest that in future collection you have to concentrate more on selecting small millets across the country and enrich your collection in the National Bureau of Plant Genetic Resources. I think special emphasis can be made to improve germplasm lines from different countries, and you have to make sure that this year you should import all these small millet germplasm lines from different countries. INR and NBP jointly should do or should work on these particular efforts. Job organized registered in this particular area are also very less. Now I expect that more emphasis should be given to the specific the characterization of these lines or specific trade published those and registered that material in the NBP more matters. Similarly, job evaluation need to be done on artificial screening, which I think last presentation was made from IIMR, and I have seen an excellent facility of evaluating thousands of journal lines or last visitors at Set with Dr. Rajan's facility. So my request and advice to IIMR would be, you have to have extensive collaboration with the Act. And I was surprised to see that when you showed different lines which are resistant to blast across locations, there was no line which are highly resistant. But since this particular trade is controlled by single G, and I am expecting that you will definitely get variation for blast resistance, some lines which are highly resistant in all those lines in your crossing program. And for that, artificial screening is very, very important. But the field conditions, many times there is a problem of other stresses which kills your organism and it kills your host, and it is very difficult to make out whether it is killed by disease or by the other traits. Therefore, artificial screening of all those lines which you have moderate resistance or highly resistant under field conditions is very, very important, and use of the protocol developed by Set. So these are some of the points which I noted during the presentation, and I am sure that you will make these as a part of the proceeding. My suggestion to all the participants would be that we should take part in the discussion, and as told by ADC, you might have discussed during your preliminary meeting which was already conducted, and a strong technical program has already been made. So your participation is very important in all these sessions for discussion and suggesting some new ways of improving this particular program. Thank you very much.
Thank you very much, sir. Thank you, the chair and co-chair, for conducting this particular technical session in very nice way, and that too in time. And special thanks to the co-chair who has conducted the proceedings very perfectly. And again, my special thanks to Dr. DDG sir for his recommendations, and all his recommendations have solutions in it. Thank you very much, sir, and the Millets fraternity is very much indebted to you. And all the presenters, as he told, is very nicely presented. I thank all these presenters too. And I request Dr. C. Tara Satyavati, Director, ICR Indian Institute of Millet Research, to come onto the stage to honor the chair and co-chair with a memento.
आपका जो सर
मैडम प्लीज honor the co-chair, Dr. S.K. with the memento. Thank you, madam. Thank you, sir. And thank you all the speakers for giving this nice presentation. And that ends this first session of crop improvement. We are breaking for lunch. And all the VPs are requested to go to the guest house for lunch. Honorable Vice Chancellor Madam is also joining there. And all those delegates are requested to go to the same place where we have taken breakfast for the lunch. Lunch is arranged there. Buses and vehicles for conveyance already there. So you can go into the bus and the vehicle of conveyance to the place, particular place. Thank you very much for the rapporteurs, Dr. P. Sanjana Reddy, Dr. Y.G. Bhavan and Dr. G. Girish for making all these proceedings made ready. And one important announcement is there that your sponsors are there and yet to collect the receipts are requested to go to the reception counter to collect the receipts. The sponsors who are given money and yet to collect the receipts are requested please to go to the reception counter to collect the receipts. And the coupons for accompanying persons. The coupons, one important announcement please. Coupons for accompanying persons is also available at the reception counter or the registration counter. Please go and collect the coupons. Coupons for accompanying persons. The food coupons for accompanying persons is also allowed with the reception counter. Please go and collect. Once again, I request all the delegates who are standing outside to please come and occupy your seats. If you can come slightly front, that would be very good. Who are sitting at the back can come and occupy your seats in the front. So we are going to start the next session, the technical session on crop action. I request the chairman, Dr. Lakshmikant, Director, ITR, Vivekananda Technology Education National Sansthan, Almora, to come to the dais and occupy the chair of chairman. Dr. Lakshmikant sir. I request Dr. V. Chandrika, Professor and Head Department of Agronomy, SVAC Tirupati, to come and co-chair the session. Please occupy the dais. Dr. V. Chandrika, madam. In this particular crop session, we are going to have three development speakers. The first speaker is Dr. P.S. Shekhar, who is going to speak on agronomy and physiology of all millets. I request Dr. P.S. Shekhar sir to come to the dais. I request Dr. B. Gangaya, Principal Scientist at ITR IIMR, to come to the dais, who is going to speak on agronomy and physiology of sorghum. Dr. Gangaya sir, please. I also request Dr. T.S. Sugania, madam, to come to the dais, who is going to speak on agronomy of small millets. Madam Sugania. Again, I request the participants who are standing outside may please enter the hall and occupy the seats. We are going to start the session. This particular session is on crop action, and we have three speakers. The chair and co-chair are already in the dais. I give the mic to the chair for further proceedings. Dr. Lakshmikant sir, please. One small announcement that physiology of formula will be presented by Dr. Bishnoi. I request him to come to the dais.
afternoon.
The rapporteur of this session is Dr. R. Swarna from IIMR and Dr. Ashwatthama from Vijayawada. Thank you, and I hand over the mic to the chair to further proceedings.
Thank you, Dr. Jalali. Good afternoon. After the delicious lunch. And first of all, let me thank Honorable Lady sir and Dr. Tara of Millets for giving me this opportunity. And I see in the audience Dr. Pradhan Lady, FSC is there, and all the delegates from all over the country. I've been asked to do a job for crop production section, and I am by profession I am a plant breeder. So all are safe. But my coach here, Dr. B. Chandrika, is professor at agronomy at this university. And what we do is that 1 hour time is given to us, and already 10 minutes are gone. So maybe only 5 minutes presentation and 2-3 minutes for discussion. And I will request, before I proceed, I will request Dr. Chandrika to give her remarks.
Good afternoon, Manila. Thank you, sir. And I thank the organizers for giving me this opportunity. And I am working as a professor at the Secretariat College, Tirupati. And I welcome you all to this prestigious university, Tirupati. And now we will start the crop election session. As Sir told, Chairperson Sir told, we are having 4 speakers, and each speaker will be given 10 minutes for the presentation and 2 to 3 minutes for discussion. Thank you, sir, for giving this opportunity. And I welcome all the delegates and also the speakers for this session. Thank you, sir. First speaker is Mr. Shekhar. Dr. P. Shekhar will be presenting agronomy.
Good afternoon. On the dais, Dr. Lakshmikant, Director, Asia Almoda, Dr. Chandrika, HOD, Department of Agronomy, सामने विराजमान ADG प्रधान साहब and सभी साथियों, जैसा कि in case of agronomy, I am presenting the reports on the behalf of Dr. Anil Kumar. This is some agency work, so I will present the work done by the agronomy section of the Agri on Palm Millet. So in total, we can say in summer and as well as in Kharif, total 41 trials were allotted to the center, different centers, for centers. Out of the remaining conducted, so 100% success was next. The area of the research in this agronomic section is contamination, in which we are superabundant polymers and mulching we are using for that one. Nutrient management, nitrogen, phosphorus optimization and balanced nutrition through micronutrients. As you know that the soils of the different states are deficient in micronutrients. Resource conservation technology in which the tillage manipulation as well as the weed management is the concern. Crop management, quantification of the contribution of different production factors. As you know that production factor play a very important role for the enhancement of the production. So we are also doing this research on that one. Organic and natural farming possibility in the environment. Organic manures and cow-based formulations application next. These are the experiments we have conducted during Kharif 2023. That is PMT1, response of permanent advanced hybrids and population entry to N levels. PVD 1B response of advanced medium and late hybrid synthesis to nitrogen levels in zone A. PVD 3 enhancing the biodiversified or non-biodiversified polymer hybrids productivity and quality through micronutrient under the irrigated situations. Contribution of production factors to the yield and economics of the polymer. Next. These are PVD 6 response of polymer to split application of nitrogen at different growth stages under irrigation conditions. And PVD 7 productivity of polymer either based on the master or 60 cropping sequence as infused by the organic conventional farming. Next. This is the PVD 1B where the medium or late hybrids are tested in its KB. What you at is the control ones, but in which maximum grain yield 34.52 quintals per acre and silver yield 78.37 quintals per acre yield was recorded with the application of 90 kg nitrogen, and it produced 40.2, 18.3 and 6.4% more grain yield. Here, store yield is 26.2 and 4.2 and 4.2% higher over the application of 0, 13 and 60 kg nitrogen. Next, in next year, enhancing the bioproductive and non-bioproductive polymer hybrid productivity and quality through micronutrient under irrigation situations. The objective is to find out the effect of micronutrient on the crop growth, productivity, quality and economics of the biofortified and non-biofortified polymer hybrids under irrigated conditions. Next. This is in which we have applied different micronutrients in which T9 where zinc support 25 kg per hectare plus FeSO4 20 kg per hectare plus 0.2% borax spray give maximum grading as well as the negative and where the basic ratio is maximum under the T5 where the only foliar spray of your FeSO4 is there. Next. These are the effect on the quality, efficiency, grading as well as the negative is maximum under the T9 table where we have live zinc, FeSO4 and borax for 2% as foliar spray, and we have all observed that grain contain the zinc and Fes also higher in this treatment. Next. Similarly in B zone, that A1 B zone also this same treatment is given grain in 30.43 quintals per acre, which is 31.9% higher over the recommended dose of fertilizer, nitrogen and soil, and similarly BC ratio and FeSO4 and zinc contained in the grain is also higher. Next. These are the recommendation on the basis of 3 years spending on zinc on zone 1A1. Results of both the locations, that is your Bikaner and Jodhpur, that among the basal and foliar application of the micronutrient, the treatment general support 25 kg, FeSO4 20 kg, and 2% borax was found and gave 32.2% superior over the grain yield and 21.7% higher in grain yield and 13.3% in the store over the control. The application of general support as basal application and FeSO4 as a foliar application improved the grain yield by 20.6% and 12.9% as compared to the recommended dose of fertilizer. The application of amine, copper and boron did not bring any significant improvement in the grain as well as the stability. Next. Similarly in B zone, the same treatment yield higher yield which was 34.2% higher over the control in where the gross rate is also 7,324 rupees per hectare, and net rate is also higher in this treatment, and also found highest in this treatment where BC ratio is also maximum. Next. This is the contribution of production factor to the yield of the yield and economics of the targeting A1 zone. It was observed that the median of 3 years in A1, the maximum reduction was of 64.1% was observed. Reduction in gradient was observed when irrigation was not applied. We can say if we are applying not applying the irrigations, then 64% up to 64% irrigation might be there. Irrigation was not it was followed by the without readings, or we can say in reading checks 38.0% reduction was observed. Without fertilizers 35.7%, and without cleaning and gap cleaning 35%, and the grain yield reduction among the treatment was between 12.8 to 19.3%. Next. Similarly, in A1 and B zone, the data says that the maximum reduction of 45.3% was observed when recommended dose of fertilizer was not applied. The reduction was 36.1% when weeding was not carried out. The grain yield reduction among the other treatments was between 8.5 to 43.7%, and the net return was 15.3 to 92.5% across the treatment. In zone B, revealed that the maximum reduction of 48.3% in the grain yield was observed when recommended dose of fertilizer was not applied. This was followed by the without weeding and oiling 46.2%, and without thinning and gaping 29.5%. The net return reduction was from rupees 7,296. Yes.
5 minutes only, sir.
Only 5 minutes. Yes. So in this treatment also, we will reduce the we can see in net return and distribution. Next. This is responsible for to split application of nitrogen in this A1 zones. 3 split application of, that is 25% at bases, 50% at tilling, and 25% at boot stage give maximum grain storage. Next. Similarly, in A zone, the same treatment, 25% bases plus 50% at tilling plus 25% at boot stage, recorded 24.4% to 6.2% and 10.6% higher grain yield over we can say S1, the entire dose is applied at soil, S2 50% at basal and 50% at top dressing and tilling, and S3 50% at soil and 50% at boot stage. So if this treatment is also give high yield. Next. In zone B, among the split application of nitrogen, the grain 25.9 quintals per hectare as well as storage 50.49 quintals per hectare was lowest in S1, whereas the grain yield was in the range of 27.26 to 27.67 quintals as storage 51.63 to 54.64 quintals in the S1 to S4 treatment. Next. There were DMT7 productivity of millet. Thus, local sequence in this mustard and chickpea is taken. Next. In this we have observed that the cow-based formulation gave equivalent to, we can say, 8 parts with the recommended dose of fertilizers. In where the maximum yield was reported in the recommended dose of fertilizers, and similarly nitrogen is also high. Next. Chickpea is also reported maximum in the same treatment, but it was at par with our cow-based formulations. Probably still experiment on only 2 years, so next year the information comes out. So this is the data of the 2 years. We can say productivity of permit under A1, A1, the maximum yield was reported in recommended dose of fertilizers, but it was a part, we can say but it was lower than our recommended dose of fertilizer in cow-based formulation. But the quality of the soils as well as the other quality is also improved by the application of the cow-based formulations. Next. Similarly, with respect to organic and natural farming on the microbial count, so we have observed that the cow-based formulation has maximum, we can say, bacterial count in 10 to 9 cubic formulation per unit per gram of soils, which was at par with the recommended Next. Similarly, in the workup on the A zone, this cow-based formulation is lower than our recommended dose of fertilizer, but we can say there is improvement in the seedling as well as oil content. Here the organic and natural farming on productivity and economics of the farming during Kharif 2022 and 23 in Zone B, which we have observed that the maximum return or grading return was observed in the RDF, but it was closely followed by our cow-based. Next. These are the different we can see bacterial count and fungi. Next. These are also the grain and protein content in the same. We have observed that the protein content and our yield is also at par with the recommended dose of fertilizer. Yeah, this is the plan of work. You can see we have framed the 5 experiments, PMT5, PMT1, where this is the mandatory for the all centers, because whatsoever the variety comes, or you can say entry comes for promotion of that entry, we are conducting these trials, and these are the treatments. Application and different varieties we have taken, MH 2672, MH 2673, MS 2675 and MS 2678 with the check of the SSV 67 improved. Next. For the zone A, this is MS 2709, MS 2712, MS 2717 with the check of 86 and 86 and KBH 108. Next. For the B zone, we have different dose of nitrogen with the varieties and MS 2682, MS 2705, MS 2770 with the as a control, plus 86 and 01 as a control. Next. These are our observations. Next. The resource development for irrigation of nano urea and nano DAP also. Nano fertilizers that is urea and DAP. Next. This is for the DAP. Next. This is a introductory study in the iron millets in Badal millet. This is your experience where we have to see the potentiality of the different iron and millets in the different communities. Next. These are the business analysis. Next. This is the only PMT7 is now it is PMT5. Thank you, Dr. Sir. Any observation on the presentation made by any observation? Agronomic group people, do you have any observation about experiment which is happening around the next year?
Thank you, sir. Dr. Sasinara, Director, Aurora, Dr. V. Charika, Dr. Economy, degree, geopolitics, I'm going to present on archaeology data system. What is this for? Second one from more experiments were conducted for heat tolerance by identifying the parental line of particular suitable for summer conditions that you can see in Jamnagar, and then PPM by 2B characteristics for road tolerance to identify parental lines of particular suitable for three conditions that you can see in Jaipur. And then 34 processing rate, partitioning and demobilization in the power related and related mechanisms that the location Jamnagar and Jaipur, and then overall identification of physiological rate for road tolerance in power related and related mechanisms Jamnagar and Jaipur. So I would like to inform that more I can remember of earlier time and also, but due to the and the geologists get promoted, so these experiments were not conducted and even shown here, but my data were not reviewed and technically I got order to make the PPT, so I have only the PPT, so this is the experiment here. Next, these are the well status. Next, the already experiment. Next. Yeah, here you can see the 18 entries and the Jamnagar central location. So that here you can see that relative water content was at entrance higher in 305 BCS 42 and at 65 days after showing 309 BCS 22. Then chlorophyll content was higher in both the and at entrance and 65 days in the 309 BCS 22. Next one. Then chemical initiation was also, in only 31 days in MIR 1259. Then 50% clothing in also in same varieties. Then number of dealers was also higher in same varieties. Then maturity was only 71 days. So this performed better. Next one. Then yearly attributing characters, if we go, then that 309 BCS 22 gave maximum yield at 43.23 quintals. Next one. In another test, if testing index we will go, then that highest in that 306 BCS 22. Next one. These are the conclusions: 309 BCS 22, PPMI 1309, PPMI 1307, and MIR 182 line perform better under high temperature conditions in summer. Then MIR 1259 shows character of early chemical initiation as well as maturity also. 309 BCS 22 give maximum yield under the high temperature stress, and MIR 1717 give maximum powder yield. Next one. In second experiment, PMPH by 2 weeks, there was 20 entries and spacing was 16 into 10 to 15 centimeter, 2 rows, 4 meter length. Next, chlorophyll content was higher in PPIM 1312 at emphasis and PPIM 1312 in 65 days. Next question. That is physiological, that chemical initiation was 1387-309 as 22 in 32.51 days than mean, and then seed percentage setting was also PPMI 1310. If we go the agronomic yield attributing characters, then PPI 120401 gave maximum 23.65 quintal yield. Next, then conclusions. PPI 20401, PPMI 1310, MIR 11116 and MIR 701 lines perform better under low tolerance conditions in the creek. Next. In experiment third, both the locations experiment were conducted and entity were tested. that plant height was maximum in the mean in community 158.56 and physiological maturity stage 215 centimeter. Leaf driving was highest in India 1200. Then grain yield back. If we go to grain yield, heavy is 1200 give maximum yield at 141.14 gram per plant. That's right. Conclusion pattern of estimates and interviews and the maturity was observed that maximum estimate move was found in stem, leaf and pedicle and emphasis stage, where the maximum estimate movement was found in pedicle, stem and leaf. Pusa composite 612 PM and MS17 and ADH1200 perform better for photosynthetic partitioning and the mobilization in permeate under and-fed conditions. Next. In both experiments were also conducted at both the locations. Next, 14 entries were tested. Next, here the chlorophyll content was also higher in the MPS MS70 than mean value was also higher in at 60. If you go to maturity level, then in HSB 197. Next one. Next. And go to conclusion directly. Yeah, number of leaf, number of pillars, leaf private, PDR, RDR, increase significantly from 30 to 45 and 60 approximately all the data, but leaf rate was decreased at maturity. Plant life, private and financial rate increased significantly from 45 to 60 approximately at maturity. All the data, PDR decreased significantly from the 60 area of maturity, and same pattern was formed in RDR at all the centers. Next. Thank you very much.
Thank you, Dr. Vishwari. Any observation on his presentation? No one. Thank you.
Thank you very much.
Now the next presenter is Dr. B. Gagaya. And Dr. Sir, you have 12 minutes time.
Esteemed Chairman and AGA participants, I shall be making the presentation about the progress made during the last year in agronomy and physiology. Next one, please. We are having around 16 agronomists working both on forage, grain sorter during Kharif and Rabi season, and 6 physiologists working in physiology. Next one. If you are looking, we have conducted 15 agronomy trials. And if you are adding centers, they will add up to 70 trials. In physiology, we are having 6 trials during Kharif and Rabi on different crop profiles. During the last year, we have seen cyclical moisture stress during the Kharif season and also continuous stress during the Rabi season. Most of the signs are moderately saline, moderately operate, non-saline, growing, organic carbon negative. This is the moisture stress. We see most of the stress during the Kharif season as well as the Rabi season. The Rabi season stress is more. Next one. So if you look at, these are the acre trials under AOT. We have conducted, these are the single-cut forage sorghum hybrids. The 3 sorghum hybrid, the varieties which we have new entries, they perform best at research center over the chairs. And at Pantnagar also we are having one entry SP278 performing best for green fodder and also some Indiana we see and dry fodder also they are performing better. In multi-cut sorghum we have evaluated one hybrid against 2 checks. It is promising to the checks. Next one please. In Kharif grain sorghums we have found positive results in terms of grain yield for these yellow sorghum entries. Compared to that one, we are having the new entries performing better, and also we are having one hybrid that is performing not superior, but these yellow sorghum PPV yellow sorghum varieties, there is promising results in terms of green yield, and economics will be in similar lines. This is a sweet sorghum. One hybrid is evaluated against 2 checks at Indore, Parbhani and Udaipur. And also we have good training with the removal analysis and these are the regulations, it is made, it is relocations into the department. These are the regulations evaluated with the redundancy season, is compared with the check and it is pronounced already. And the grains organs, colorless organs are compared with the grains organs are compared, grains organs are the probability, is there. And the is a Product is a important commodity that produces both leading product as well as the green seeds that are going commodity produces in product also. If you are growing product with 150% fertilizer and irrigated conditions, best product and other mixes are possible. And also acidic irrigation is having higher water limits because of its strong structure. If you look at the Rhoda production, we are producing the both Rhoda and grains sorghum and organic production systems. We see if we are applying RDF through vermicompost and biofertilizers, the yields are best. This is the fourth year of study and we are getting the yield losses due to fertilizer application over non-organic manures is getting gradually filled up. We anticipate by the 6th year the organic is going to be as much of inorganic fertilizer. In organic grains sorghum productions, we have the rabi sorghum we are having across centers. Here we found that if we are able to apply vermicompost along with biofertilizers, we are getting the best yields. Next please. In Rhoda, the harvest season is very short, only in January we are getting. We wanted to extend the availability of Rhoda grains sorghum, so we have taken up the extended windows of sowing. Here, urban India extended is going up to probably 15 December, but normally the late sowing is good yields, but the yields are very reduced. This is the process of we tried annual DAP and the replacement is not giving promising results. Like reduction in yield is we are applying foliar fertilization. With respect to rabi sorghum, we are applying all the nutrients, NPK and also iron, zinc and boron. Significant yields are possible in grains sorghum during rabi season because nutrient uptake through soil is hampered and foliar application is the way to enhance the productivity in rainfed conditions. And the foliar in rabi sorghum we have tried the foliar nutrition and here also if nano urea spray at PA and chemical stage, initiation stage or NPK, Ketan and iron spray they are having a yield advantage. We are yet to confirm. This is the boron nutrition. We found that 0.1 ppm boron spray will give good yields and the yield gain is because of the pollen viability enhancement and also percentage grain filling. This is the intercropping. We wanted to enhance the millet productivity across the country. As area is not coming, we have tried the millet intercropping system. There is yield advantage up to 60% when we are combining the millet with millet cropping system. This is evident from land equivalent ratio. You see even sorghum bajra is having higher. Next one. Sorghum bajra is around 1.44% yield land equivalent enhancement and this up to minimum 15% yield gain. Land advantage gain. Next one please. Summer we have initiated our summer last year. 15 units are evaluated. We found that the two Kharif 26 is 296 and BK broadly and among the rabi seasons and winter these are the rabi season they are summer season and they perform for the yields in terms of the disease rotation. We are going to extend this trial to all other centers. Next one. If you want to physiology, this is a system from generalized development evaluated for performance under saline conditions where these are the biomass production is not hampered, but seed production is significantly reduced for seed sorghum. If you look at the foliage sorghum types, both foliage as well as the seed production is hampered. In seed sorghum, you are able to produce high biomass, but seed production is not possible. But in fodder, both fodder as well as its grain seed production is hampered under salinity condition. And this is the very good contribution from physiology group. Climate change is hampering the productivity of millets. Now we have seen that by what way you are altering the sowing windows, can you able to compensate the yield losses. We have found that both arboreal and Akola, if you are advancing the sowing dates, the yield, there is productivity can be enhanced and the productivity loss can be overcome. This is sowing. Next one. If you come to you can manipulate the variety also. BM35-1 when you are replacing with Foolish Vichitra at Akola and Ravari, and also BJ144 at Vijaypura. Variety replacement and also the population, lower population is performing better under these situations. Regarding previous one, there is a three-way interaction also. You can go these rods and the lower population, increase sowing and also the reduction of variety, you can enhance the production. And the climate change impacts that you overcome in a major extent. On the other hand, you are having higher risks than the current situation. Now, we have already discussed the technical program. These are the ability trials next one. These are the ability trials next one. This is the new trial we are proposing as per the direct irrigation. We wanted to assess the water productivity, especially in this program, and also soil fertility changes across the urban regions. For this, we are methodology has been given. Next one, please. This is the ongoing trial. Next one. This is we are adding new sectors for the irrigated trials. Next one, please. This is also ongoing trial. Next one. This is solar nutrition. We wanted to reassess the trials for one more year. Next one. The Murudas of them we are continuing with organic production system. Next one. This is the Murudas sowing window. We are continuing for one more year. Next one. This is the growth sorghum intercropping system. Second year we are confirming this trial. This trial is introduced in Kalminet also based on our research findings. Next one. This is the ongoing trial with the urea nutrients foliar spray. Next one. This we are concluding this trial. We have found that 0.1 ppm boron spray is promising for Rabi sorghum. All the centers, sector, Mandal and Tandur. Next one. This is physiology. The new trial is we are assessing the nutrient use efficiency and responsiveness of the genotype level at physiology level. Once the physiology material is identified, then we will enter into the economic materials. This is the drought screening. We are screening the selected genotypes of germplasm under shallow soils at Vijaypura. Next one. This is other trial phenotyping sorghum in the root structures at Vijaypura and the others at the next one. This is, we are modifying this trial with the climate change. Whatever the best results last year we have received, we are modifying the technical program. And this is ongoing trial. Next one. This is also ongoing trial. Next one, please. So this is the trial we wanted to confirm it again for the second year, foliar nutrition with physiology. Next one. We are concluding this trial where we last year we have conducted cotton intercropping with sorghum genotypes of different genotypes and we have followed up the results. Next one. We are also concluding the boron nutrition of the sorghum under physiology. So this is the technical program. Thank you very much.
You are ahead of time. And any observation? No. Then just Madam one study you have from 4 years will be composed plus that. I hope that reputation is coming now.
Converted organic farming within 3 years study, right? Now it is first year of organic. So this trial will continue for another 4 years.
And what good information has come that lower population plus early sowing and right variety that can mitigate the climate change.
In Rabi we are sowing growing sorghum under receding soil. That is why lower population.
So then all the breeding centers have to test their varieties under these conditions. Because variety has to be identified, which one is suitable. Then this one trial you told concluding with that boron, so that will come as a recommendation. And that experiment which you have planned for drought screening, how they will be ensuring the drought?
Sir, it is a shallow soil, less than 45 centimeters, and receding soil. We are having soils at Vijaypura. Yet the soil depth is hardly 45 centimeters. So automatically the drought is there and moisture storage is less.
I don't know.
The rainfall itself is less than 50 mm or 50 mm.
But the change climatic conditions, we are getting rains where there were no rains and we are getting heavy rains when we are not expecting the rains.
We reserve the rainfall received and also in shallow sites screening. If the rainout shelter will be insufficient, we don't have that.
But somehow you have to ensure that drought is there. And this sorghum plus cotton again it is coming as a recommendation? Okay. Thank you very much. Now I would request Madam T.S. Sotinia for small minutes. You have 12 minutes, 3 minutes for introduction.
So we have 14 centers, out of 14 centers, 4 centers are voluntary centers. This is diverse climatic soil types. We are having soil from slightly acidic to alkaline soil and organic carbon from 0.4 to 0.7. So and we have nutrients low to high. So next. These are the production constraints. So based on the resources available at different locations, we have planned experiments which can provide solutions. So these are the production constraints. So based on which we have formulated the experiments. Next. So these the abstract of agronomy trials which are conducted last year. So under breeding trial we have conducted 5 pre-release genotype trials, different fertility levels, the nutrient management, here organic farming in small millets, then nano fertilizer, foliar application, then screening of cultivars for nitrogen efficiency. Next. So weed management, so moisture conservation, since these are more under arid and semi-arid regions where moisture stress is always there. Then next, millet-based cropping system to overcome the risk and to enhance system productivity. Then evolution of retomography, the first kind of experiment in millets we have initiated last year. Then performance and economic evolution of formations. So these are the concepts we worked during 2023. Next. Next. No. So under breeding trials we have conducted trials on Pingo millet. So 2 test varieties against 2 checks. Here the 100% RDF was found. The varieties but they didn't show the response for either higher or lower nutrient level. So whereas in fasted millet also, so the 100% recommended dose of NPK was found sufficient, and also the test varieties failed to exert any significant influence compared to the national check. So these are the some of the field trials conducted at one center. Next. No. Can I use the PDF, sir? PDF. time. So in finger millet also, where I have already explained the foxtail millet, then Proso millet. The Proso millet varieties, they are shown response for a lower fertility level, that is 75% RDF was sufficient. Here the test variety has shown significant influence over national checks. Next, little millet, here also 100% RDF was found sufficient, and even the agronomy efficiency, if we calculated, that was found economically feasible at 100% RDF. And with respect to the pre-release genotypes, the test variety, that is LME 564, has given higher yield when compared to national checks. Next, ground up millet also, the lower fertility level was found with higher performance of the crop. And even among the varieties tested, the test variety was found with higher values of yield when compared to national checks. Next, these are the pictures. So next, the organic farming. To know the impact of organic farming on small millets, whether it is feasible or not. So we have initiated this particular treatment, finger millet and poro millet, last year. So here the treatment where it was imposed with RDF, the recommended RDF was found with higher values and compared to other organic practices at the end of 1 year. The same result was obtained with poro millet. So the vermicompost, where vermicompost was used to supplement the nitrogen requirement of the crop, it was also found with good values in comparison with the RDF. Next. So with regard to nano fertilizer, we have initiated this trial 2 years back. Here the 75% RDF along with the 4 years way of nano fertilizer at 2 important critical stages was done with good values and supported with the economic indices, and also the nutrient, especially nitrogen efficiency and uptake is also better with this particular treatment. Next, so 14% and 10% in enhancement, and the cost of production is also found lesser with this particular treatment. Next, foxtail millet here also the same treatment has found with good result. Next, so the little millet. Again, the progress way of India at 2 important stages had good scope in enhancing yield and further reducing cost of production to 7%. Next, we have conducted this particular trial with finger millet cultivars and nitrogen efficiencies, and nitrogen is a major nutrient required for the millets. Here, we could find the differences among the varieties which are found manageable. The difference with respect to nitrogen efficiency, this was found. Next, this is the experiment conducted, and nitrogen efficiency was also found among different varieties tested. Next, the method for mulching with nitrogen, this hydrogen is a polymer, natural polymer, and a super absorbent material which can imbibe water, absorb water to 300 to 500 tons, and it also the principle of this osmotic pressure. So this was tested along with mulching and the impact of this particular mulching and nitrogen on performance of the crop and also on the soil properties. Here, among different treatment combinations, the crop seed merged with 5 tons per hectare along with 5 kg nitrogen. So this had very good scope in enhancing yield, and also the gravimetric soil moisture content. Wherever we use this combination of residue along with mulch, the gravimetric soil moisture content was higher than compared to other treatments, and even the water use efficiency also found enhanced. Next. So you can see with this figures and also the important pictures we have when we conducted this particular trial. Then, so this particular experiment was also conducted. Next, for foxtail and even little millet, there also the combination of mulching and nitrogen have scope to enhance the yield and further improving the soil parameters, like even the microbial volume also found enhanced, and even the enzymatic activity was also found enhanced in these particular treatments. Next. So this experiment was also conducted in Barnett. There also this treatments had important results. Then returnability, just to know the impact of returnability in fingernail, whether these millets can be suitable for returnability. So this particular treatment experiment was initiated last year. Next, here three factors were tested: stage of harvest, nitrogen levels and height of harvest. So here among these factors, nitrogen levels had found the significant with respect to performance of the crop. Next. Then last experiment, that is performance and economic evolution of farm machinery. Here we compared with manual combined harvester along with reaper with manual method. Here also whenever we use this combined harvester, there was around 90%, 98% labor consumption is reduced when compared to manual, and also 25% labor consumed when compared to the reaper. So even the time consumption also was reduced when we overall this machinery harvesting. Then next. So this experiment we are concluding that the water soluble fertilizer here 75% earlier along with 2%. Mainly important critical stages of culinary and biological initiation was done good with respect to performance of the crop. Next. So here the benefit cost is particularly economic analysis. So that indicated that there is an increase in the benefit cost ratio. If you use this 90 after reducing the 25% earlier. Next. So these are the supporting graphs. So uptake of NPK also enhances this particular treatment. So this is the outcome of the trial. So the 2% 90 watt at development critical stage can be found as effective technology for increasing the productivity of the crop and also for reducing the cost of production. Next. So your chemical limit studies here we have conducted all the emerging herbicides along with the post-emergent herbicides, knowing the efficacy of these herbicides. So among different herbicides tested, the field, so that is standard practice needed more. So comparatively among different herbicides, so the T3, that is the Mesothelium at the rate of 5 kg per hectare. So this was economically feasible. Next. So weed control efficiency also further enhanced, and also the weed index also considerably less in this particular treatment. Next. So then also next is the full data. So here also same result was obtained over the locations and over the years. So we can see the weed control efficiency is considerably higher in this particular treatments of T3 and T7. Next. So if the farmer wishes to go for integrated aspect of weed management, then we can suggest the post-emergent application, early post-emergent application of this hormones, that is methyl-methyl plus chlorine at 20 gram active ingredient per hectare for the effective management of weeds. Next. So we also tried with little millet and same result was obtained with this particular crop. The next. So this is the full data. Next.
For example, because the really response for the lower fertility, like lower fertilizer, then we can recommend the reduced.
I would suggest you can analyze last 10 years data. You will find similar kind of result is there. So better to explore other areas also. So Dr. Shekhawat, I think there is one recommendation is also coming from that. Since he has left, you may note it down that one recommendation is coming. So from all these presentations, some recommendation will be coming. I hope the reporters will be noting it down and those things will be coming as a form of recommendation. Already 3 years has passed, so those will come as a recommendation. So with these few comments, and madam, one more comment. This how big is the chemical weed control trial is here? Okay. So with this, I hand over to my co-chair.
Thank you, sir. And I congratulate all the speakers for their excellent presentations and good deliberations at almost all the centers, and millet and sorghum and small millets. A good research is going on. And with regard to this small millets presentation, madam, I have some doubts regarding sir has talked about the weed management without bensulfuron methyl plus tetrachlor. Is it showing any phytotoxicity or is it ready mix or pre-mix, and having some thing about that? And what's the
reduce the dosage pattern. So this recommendation is 5 kg per hectare. Usually it is 8 to 10 kg for rice and all. But here in small millets you have tested and made formulations. So 5 kg per hectare we tested. And then it shows for cytotoxic effect.
Is it, are you using as pre-emergence? Is it ready mix, madam? Pre-mix? Pre-mix. You are using as pre-emergence only. But little millets, mostly the millets are susceptible for these herbicides.
That is why we get to the dosage.
Means it didn't show any phytotoxicity. We have not
At
recorded.
this dose we didn't find the phytotoxicity.
It's a good observation. And one more thing regarding hydrogen. What's the cost of this and is it economical? At what dose? 5 kg you are telling? Is it economical and where it is available to the farmers?
So from the point of soil fertility and its microbial biomass, the indirect benefit will be there. And crop performance of soil, so moisture availability. So it works on the principle of osmotic pressure. So whenever there is a dryness, then it starts giving the moisture.
Cost
is 800 rupees per kg.
800 rupees per kg.
So yes.
Economics.
Yes. That is what, madam, 2.55 and 7.5. So even we can use 2.5 kg if you want to have more advantage.
Along with the small millets, yes, was not much economical and we have to offer the economics also. And we have coupled with crop mulching.
Yes,
mulching.
Any land residues.
Many more advantages so that we can reduce losses. And one more observation regarding organic farming, so we are recommending vermicompost integration.
No, madam, it is actually the second year. So we need to wait for 2 more years to know the actual impact.
But only vermicompost
it gives the nutrients. So that is why the treatment had more beneficial.
But if the farmer want to purchase vermicompost, it is more expensive than
organic farmer.
Yes, the economics has to be
for organic farmer.
Yes, the farmer will prepare himself, it's okay, but it is okay, but there is economic one more thing, nano urea it is advantageous. Nano urea each year it is established. Nano urea cannot, or it cannot substitute fertilizers.
That is why we are continuing this experiment. We are not concluded.
And there are other presentations also, sir. Good recommendations regarding the climate change, COVID reductions, and some intercropping and organic farming. And another observation, sir, regarding the village practice in village, sir, village special advantage with
Madam, the area under village is already very low. Some of the area which is accumulated is in coastal Rajasthan. The solar is Karnataka. If you go for the intercrop, you can bring in blue crop. Also, there are sometimes more economical than the existing. In this context, we wanted to enhance the area under millet crops. By millet, those are cereals. Our objective is to enhance the productivity. You see the example of sugarcane, wheat, grain, both are cereals. The objective is higher productivity. Here the objective is to enhance the millet, also the fermentation, blue crops,
initially competition with this sir. Good regulations, sir. Congratulations on the speakers. Thank you.
Thank you, madam. I congratulate all the 4 presenters. The presentations are excellent. And only last thing I would like to say is that whatever you do, cost you have to finally see. Otherwise, it may be very good, but farmers will not adopt it. And again, this millet, you can rethink it. Expanding the area is okay, but at the same time, you have to look for sustainability also, because they are heavy feeder crops, and you will be exhausting the crops, and maybe the next season till farmers will not be able to take any crop. So that kind of thing also. You are an agronomist, so you know better than me. So you have to take care of those things. With this, I once again thank all the presenters for wonderful presentation. And finally, I would like to say that without interaction, the presentations are meaningless. I was told that it has been discussed, but even if it is discussed, many new ideas come here. So the audience has to interact. Otherwise, there is no use of having a physical kind of meeting here. So with this, thank you very much.
We are concluding the session on crop production. We thank Chairman of the session, Dr. Lakshmi Kant, Director, ICRBP Sarmona, and the co-chairperson of this session, Dr. V. Chandrika, Professor and Head, Department of Agronomy, SBAC Tirupati. We also thank the speakers of this session, Dr. P.S. Srivagan, Dr. Vishnu, Dr. B. Gangaya, and Dr. T.S. Sukanya. Also the rapporteurs of this session, Drs. R. Swarna, Ayyanar, and Ashwathama Vijayapura. Now, we request Dr. R. Madhusudana, Project Coordinator, Sorghum and Small Millets, to felicitate the Chairman and Co-Chairperson of this session.
Sir.
Thank you, sir. The next session, crop production authority, shall begin soon.
So we are starting the next session on crop protection pathology. Here in this session we do have Chairman Dr. S.S. Mane, Dean PDKV Akola and the PAMC member, and we have a co-chair here, Dr. Reddy Kumar, Professor of Plant Pathology, and we have two presentations, one in pathology of pearl millet and sorghum by Dr. I.K. Das and pathology of small millets by Dr. K.B. Palanna. I request the chairman of this session, Dr. S.S. Mane, the Dean of PDKV Akola and the PAMC member to come on to the dais, please. I request the co-chair, Dr. Reddy Kumar, Professor of Plant Pathology, to come on to the dais, please. I request Dr. I.K. Das, Principal Scientist at ICR IAMR, to come to the dais, who is going to talk on pathology of pearl millet and sorghum. I request Dr. K.B. Palanna to come to the dais to talk on pathology of small millets. So here the rapporteurs being Dr. G. Rajesha from IAMR and Dr. A. Zah from TNA. These are rapporteurs of this session. And we have three presentations here, one on pathology of pearl millet and pathology of sorghum by Dr. A. Zah and pathology of small millet by Dr. K.B. Palanna. Without taking much time, I am passing on the mic to the chair, Dr. S.S. Mane, for further proceedings.
Good afternoon, all of you. Before starting, let me introduce myself. I am Dr. S. Mane, Director of Execution Team, Faculty of Agriculture, Dr. PDK Akola, and at the same time, PAMC member. With me, I have my co-chairman, Dr. V. Bhaskar, Principal Scientist, Department of Pathology. The rapporteurs are Dr. Rajesha from IIMR and A. Sudha from Guru. And in this session, we will be having the three presentations. Pathology coordinated by I.K. Das, pathology sorghum by I.K. Das, and pathology small millet K.B. Palanna. And outside, let me welcome all the dignitaries on the dais and outside for this presentation. And I request Dr. I.K. Das, please start with the presentation. Respected Chairman, Co-Chairman, and dignitaries of the dais. Today, as we remember this family pathology trial that Samanya progress. Actually, Dr. Chennai is investigation in order to activate this program. So I am presenting on his behalf. So those are the pathology program. There are 9 different types of experiments, and that go on in 9 coordinating centers and supported by another 4 coordinating centers. And those centers are resources in different zones, A1, A2, and A1A and B zone. So these are the list of trials that include the disease screening for advanced as initial graded materials, and monitoring of behavior for downgrade as well as loss, and then management trial for downgrade
as well as blast. And there are some summer trial is also there one, and some basic research studies are also there. Next please. Now while coming to this advanced material screening, so taking this initial and advanced material, there are 259 entries, and those entries are screened mainly for the down values, mark, rust, blast and argon. So they have a fixed criteria for what should be the minimum level for promotion. So that is been given for down value, it's up to 5%, blast up to 3 on a scale of 1 to 9, and rust and smart argon it is 20%. So next please. So coming to this initial materials, this year there are 106 various materials which are tested in initial radical and heavy trials, and you see that in down value, out of the 106, 95 was highly resistant, and here this highly resistance does mean that it is highly resistant because these are done in 6 plot conditions, and the susceptible check here produces this is as high as 70%. So in that case, the level of 5% you are getting means that is the material is really having the resistance. So in that category, 106 out of 106, 95 come highly resistant and 11 come the resistant. Now when we come to the blast, here actually the infector role is used, not artificially inoculated for every plant. Here the situation is that out of 106, 94 are showing the resistant. But I would say that pressure is a bit less as it is shown by this overall mean here, and we get 12 model resistance line. And so for the argon, smart and rust, they are all in permissible limits for range of the variety. Next please. And then the advanced material, the situation is that out of the 32 are registered to down because there is a two-stage of advanced screening. That's why it is the situation. And the last we will share are registered and moral registered. And last and mark all are in final solution. Next please. Now there is an experiment to study what is happening on the release variety, whether they are after cultivating years the same level of resistance is same or they are going some degree of resistance level. So last and normally it is a study, and out of 35 entries, the release variety is an agreed 33 maintain that resistance, but 2 they came down up to the resistance level from high resistance. If it is a high resistance, they only go for the yield. And in case of last out of 29, out of 35, 29 maintain that resistance level, and the resistance level gone down. But it is not the case of argon, rust and mark, or in the same level of permissible. Next please. Now coming to the calculation of conversion diversity in the down value. Now there are 41 entities that are tested in down value variability nursery, and we got that 29 entities are highly resistant, and in across all the 10 locations, this does indicate that these entities is really having the resistance and the highly resistant, and there are some entities which are resistant, and for some about 10 locations, some about 9 locations, and some about 8 locations. Next please. Now coming to the characteristics of the location, distinct location, MRI, we see that the download and initial and these are the locations where the disease pressure is very less in spite of having the sick clot. That will indicate that the pathogen population in those locations is less virulent. And when it comes to the isolated conditions, the virulence is very high, as in the viral incidence is also very high. And next please. Now there are probably last regulatory nursery, they are also doing ancient essay, and he told that overall Vijayanagaram and New Delhi. These are the areas where that incidents, this has become somewhat susceptible, means congenial development of disease happens. But in other locations, the situation is that the mean is around the 3. So those are the locations probably not as good for natural selections or as it is for Aurangabad, Vijayanagaram or New Delhi. Next. Now some basic research also studied by Dr. Chandran. Next please. Now some first was that bacterial isolates are isolated from the panicles of this valvulate, and the objective was that whether those bacteria is able to protect the infection of the spores of the downward mildew. And they have seen that those particularly an isolate Bacillus licheri is having some promise in that case, and they have also sequenced that and they have deposited in this NCBI database, and that has also been published. Next. Now diversity of virulence genes in minority populations infecting millets. Actually, this is a very important thing, and we know that for the blast causing megamote is having a wide range of host strains. So, taking 156 isolates, they tested using different primers of different genes, and they got for the MPS1 and TPS1 and SLP1. These are the gene primers which gave more than 96% similarity, indicating that in millets, those are the genes responsible for blast resistance, and also some similarity in adrenal genes also observed, particularly rice and pearl millet. Next. Now pathogen diversity. That is the most important thing when we go for the analysis of the different locations, how the populations are distributed, because ultimately the material is to be exposed under natural conditions. So here that around 116 isolates from across the millet growing regions are tested on differential host line, and it was observed that there are 14 different types of variants available, means those you can say are the pathotypes also. So the pathotype number 13 and 14 also is highly related. Whatever now different lines are available, all are indicated by this pathogen. So these are very important, and this can be utilized for the artificial killing under laboratory conditions. Next. Now, evaluation of integrated disease management studies that could study one of non-mildew and another last. And for treatment as well as treatment was conducted taking some microalgae, ESD and pseudomonas, and overall treatment was observed is very good. Actually, it is reducing the disease incidence from 80% to 6.6%, almost the normal resistance level from the perceptual to normal resistance. Now it is in yield. But this is again a typically happen. Next, I got the last also to treatment very much counseling that includes the treatments with similar proportions, and also the chemicals like pyroxychloroquine and tetracycline. So those who are also taken competition of management of the blast. Next. Now monitoring our power field with the master that can make blast and blast, which is probably more damage to the power field, and blast and blast are becoming more in states like Maharashtra, Rajasthan, Gujarat, Karnataka, and the incidence of power field is gradually decreasing. Now it is coming up to 0 to 65% in Maharashtra. So these are some observations based on the survey details. Next. Now there is a summer trial actually to see under the summer condition how the disease transmission is. And really that it was observed that normally downwind can push some extent, what it is that is having a resistance level, it is less than 5%. But there is no other green in summer. So that is an ecology and the plant can be, this is our farm can be easily grown without any other sort of animals or diseases. Next. So these are the technical programs that have already been discussed with the groups and also in our team members and editing. So most of all the experiments will be repeated. And then we will experiment, one will study the equivalent of the beginning of accessing the blocks in the natural condition and the equivalent of acidic variations and development of differential host and the diversity of pathotypes. Actually, differential host lines is already there, but the main target is the diversity of pathogens, because after 3 years you have to carry those things to know whether there is any change or not. So next please. So these are the experiments which will be repeated next. And this epidemiological blast development is a new study we will be doing, and we will be analyzing this disease progress, how it is happening in the farmer field and in the experimental blast. Next. So these are the experiments what to be done at all the centers. So this is the routine things, aggregate, it is standard protocol. Next. There are 2 recommendations, but I don't think these are much of the recommendation. Actually, these are some multiple disease registers lives have been observed, and the application of bioagent efficacy reduced automatically. That is one of the observations only. It may not come under the level of recommendations. So next, so with this I conclude the millet part, then I move to the sorghum part. In sorghum pathology, this program is supported by seven major centers, and we are having some centers like PAU, Ludhiana, Hisher, and Agris and Vijayapur. They also support, though they don't have the regular pathologists in the program. They take from other program and they support the program. That is how we get the information from all across the southern going part. Next. So these are the distribution of the disease, and we know that Kharif and Rabi is 2 different ecology. And here in Kharif, the diseases are well defined. If you go to the north, more of the foliar disease like anthracnose, junipero spot and grey leaf spot come. And you go to the south, it is mainly the grain mold and leaf blights and some kind of puka point, and those type of diseases will come in the grain sorghum. And then go to the Rabi situation, again the state of different diseases are there. The rust will come again and charcoal rot will come again. And but leaf blight comes in both the situations. It doesn't mean that other diseases doesn't come here and there, but it comes on the predominant distribution line. Now, the experiment details are taken from basic studies like yield loss estimations and identification of new molecules for other management, evaluation of national crossing material quarter, discussing that many materials have been generated in different centers for evaluating the for the disease registration purpose, green mold registration nursery, foliar disease nursery, and integrated management insect payload. These are the one part. And this is the other part. Other part means these are the different. Sorghum is having 5 types of 4 types of product categories. So in all categories there will be initial trial and advanced trial. So that's why it comes this much. So evaluation materials will be there, and these are the centers and locations I just tested last week. Now in coming to the weather condition, because for the disease, this is the most important thing. Whether you go for artificial insemination or not, if weather is not favorable, it will not come. Another condition. So we see that this is the season when that in the upper part of India, like Udaipur, Ludhiana, Bharatnagar, only the rainfall gains from a very high amount. And when you go to the middle part, Patani, Ankara, Surat, and Hyderabad, again the rural part having very high rainfall. Others are below 80 centimeter. And when you go for the further down, the Dharwad and the this year rainfall further reduced, and it is only 80 centimeters. Now some of the things we see that it is only the Surat and Ludhiana, Surat and Patna had the high level of humidity of 80% was there. In this other location we see the humidity and rainfall, particularly the rainfall. Humidity is there, but particular rainfall is not there that much. So the proportionally in those areas, disease is also reduced. Next please. Now this situation is carried with the major diseases grain mold followed by some leaf diseases like blight, anthracnose and juvenile leaf spot. Rust also comes sometimes in Kharif. And we see that grain mold is major problems. And we see that Gujarat, because of the rainfall, the grain mold score, mean grain mold score is almost 4.5. It's a bit high for this advanced area. And when you see the Rajasthan and Tamil Nadu, it's very rare because of the rainfall. Because grain mold is such a disease which is totally dependent on rainfall. There is no much host plant resistance. It is our study of the 100% proof that it is after physiological maturity, it is only the environmental factor which controls the grain mold. That is why we are not able to contain the grain mold. But if you want to contain, you will get very hard seed that will not be utilizable. That is the barrier for the breeder. That's why this much progress is not coming for grain mold. And for leaf blight and anthracnose situations, you can see that all the state you are seeing the leaf blight and anthracnose. And other disease is not coming in all the state. That indicates that leaf blight is really increasing and becoming the pan-India situation. Next. Now this situation is very again charcoal rot and leaf blight is there and rust is also increasing. So that is another part. And rust is increasing, though it is coming maybe after flowering, but also this causes a lot of yield loss. Next. And we had some yield loss experiment by this recommendations of the ICR in our meeting that whatever varieties and hybrids yield loss estimation is there, it is very old. Now you are using very much highly improved varieties and hybrid. Why the same type of loss is happening? What your data says? In that case, we study those things and we see that for the foliar diseases, mainly for that this anthracnose and juvenile leaf spot, which is major hindrance for this forage production in North India. We see that available green fodder loss is 12.8% to 14%, and when the disease pressure increases, it goes to 28.6 to 39.6%. And available seed yield loss is also there 22 to 24%, and potential yield loss is 34 to 62. And this is the third year study. And when we sum up, we get the 3-year average. The green fodder yield loss is 15 to 35.5%, and the seed yield is 11 to 15.8%. So it's a very high loss, particularly for the seed, because seed production is very difficult. And in that case, we are losing almost 16% because of that disease only. Next. A new molecule for target, we tested two molecules from the market. People say, oh, you are targeting using fungicide, and people are going away from the targeting. We have to have some chemical. And particularly for the seed production problem, the security is not happening with the online, with the media. That is the situation target comes. So hybrid seed production is we have to develop some data of our own, because there is no available in many of the fungicides for use in the problem of millet. So all of us have to develop database that we can have some database on these, and we can say, okay, if you use this seed, we have the support data for that. And in that case, we got some temporary and hydroxyl combinations and temporary and fluoride combinations and hexagonal. So these are very effective for the control of the harvest. And cleaning that applying that in the that is the time the power and the is also on other also, great one also. So this application also controlling other diseases also. In that we also calculated the BC ratio as the environmental is having very less price, the 1/10 of the price of the other environmental. So it is getting a bit higher, maybe 3.1 BC ratio. So it is very good to use that one and for the city production law. Next. Now, even the irrigation of national cropping in India for rainwater resistance, even that one in multiple locations, and in multiple lines of gain sort of 3.8 and 3.9, and using any rainwater resistance, so you have to look on into other part also, that how much is the gaining side, and how much is hardy to not be very hard also, we will not use that one. Because the background, hardly will not taken by the farmers and all the things. Next, and rainwater resistance also similarly got the good lines, especially every 3 year one, very good, 2.84 is a very good score. And IC, NCA 8 also 2.3 is very good score, almost at par with the resistance. Next. And integrated management and mistake-based diseases, I saw that hexaconazole is experience working for the target and other diseases, so that chemical was integrated with the insect management practices. And we wanted to know the combined effect of those things, and in that case also we saw that argatine, green mold incidents, and also increasing, and stem border and that date has also reducing with the different treatments are there. Treatment number 4 is calling that much. But here the treatment is also accompanied by the 4-year application. Next. Now comes to the varietal part, means screening of the different breeders materials. Now you see that we take different materials. We see that advanced Iberian variety. Here you can see that mainly for the NDHT trial, you see that though the disease pressure is very high here, you see the grain mold in the best released variety is coming, what and the other things also coming around only 1 or 2 are there which is showing with higher score. So it shows that whatever material we are getting in the advanced line for testing in the ASFP system, not much having that much variation in that for the resistance is concerned. And the leaf line is also like that, and we got some line like SPH 2021, SPH 202628, which are tolerant to multiple diseases. Nowadays, for the millet, I will say, instead of going to the individual resistance, we should go for the multiple resistance, because individual resistance doesn't work in this case. How many type of resistance is incorporating? You have to have some multiple resistance so that directly you can use those things. Okay, for basic studies it's okay. We can see up to gene level what is the resistance on that one. But unless we are getting the multiple resistance, it is not going to work for this very low end crop. Next. And for the initial hybrid and varieties, again in the same situations, we see that these are the varieties, especially for SPB 304 and hybrid SPB 205, having very good resistance of grain mold, and also leaf light also. Next. But one thing about it, the foliar is the most important, and the monolith important. You see that SPH 2045, SPH 2040, this is very important, having less than 3 4, and susceptible to take going more than around 6. So, though it is not as required as much later as it is required, but it indicates that these are the genotype having good tolerance to that. Next. Similarly, the whole hybrid variety range, number 1 kinds identified from North Indian conditions, for Central and South Indian conditions. And now it is SPB 2884, SPB 367, and SPH 2056, and so on. The food program also similarly, there are animations and community for that is very much important for the because sugar accumulation is decreased, lead infection is there. Next. The revision program coming to that, you see one point you can see from the slide that whatever the leaves the material are using there are almost becoming susceptible. More than 25 CRI is susceptible. And you can see that the CH has become 27, CH has become 26 CRI. And many things are because over the time what is happening. We don't give any importance for the disease or any releasing of material. What happens is the 10% no, this is nothing is consideration. 5% you see other things, only average. There is no clear demarcation that this would be your cutting point below that you should not use the variety. So that point is to take care and depending on what to say that one. But it is not that because ultimately they will be using compared to the animal everything around. So this is the point that you have to take and to analyze what to do about that. Next. Technical program for experiment and will continue. This is the thing already been analyzed and this is the two new things we are starting this time. These are nano formulation or improved and biotic and aerobatic sorghum because the nano silica and nano silica is having some good role in GMP is having the calcium is having a role in stress sorghum and silica is having a role in disease sorghum. So we will start those things in the next. And another is multiple reservation for talking, naturally will be built with the level whatever legislation we are finding for that one. So this thank you for this is having on the recommendation because of this there is very seed production for what I was talking that foliar application of hexagonal or the improvement of last is showing hexagonal or fluoride and overnight after plowing effectively control the disease. So this is to be kept in mind and it is required we can apply for that. Next, thank you for giving me the response. Any observation from the audience side? If no problem is there, we will start with the next presentation. Whatever study, whatever paper I will publish, so the deviate values of the environment, and that is because of the environmental effect. I think that one or two papers is only there.
Only first class. that's simple. Instead of thinking that these are not working, at least create one together. What is happening in
Okay, our data
and last time, different areas have been identified in different populations, but the problem is they cannot be validated. So validation of the future was not done. So it is stable areas will just try to identify and there will definitely.
Excuse me, sir. Dr. Basu. Yes, sir. Yes, sir. You have done excellent work. Yeah. Yes, yes, yes. So, thank you very much for your excellent presentation, sir. You have given us so many experiments with the new initiatives using some of the nanotechnological approaches and especially bioagents. So we have done the integrated management using microbial consortium, that is Trichoderma, Bacillus and Pseudomonas. So here, how this compatibility has been studied, whether the compatibility exists, how it has been tested. Just as
compatibility with the fungicides and those things you are asking.
No, sir. As you have used 3 bioagents.
Yeah.
Trichoderma, Bacillus and Pseudomonas. Whether they were used combined or alone?
No, actually they are used for seed treatment together only. And that is, I think they might be means having compatibility, though I have not done that or just presented, but I think from my experience they must be compatible. Without that, they
yes,
cannot
sir.
be.
That's what whether compatibility is studied. And thank you, sir. Thank you.
Yes.
Respected chairman, co-chairman, experts, and all my fellow colleagues. So myself, Dr. K.V. Palanda, pathologist, small millets field. So welcome all to the presentation, that is small millet pathology progress and achievements and the technical program for the year 24-25. So this is the program by the 11 scientists on behalf of all I am going to present this. So these are some of the thematic areas where we have a program on small millet pathology. There is surveillance and analysis of diseases, evaluation of the breeding materials and their problems. So disease management, EOS assessment, and assessment of the university, and we will also generating data on genomic database, and we are also planning to have some specific molecular markers for major millet pathogens. So before going to the actual presentations, there are some recommendations in the last AGM. So these cross-infection studies of this area population which is infecting millets, and then also we are suggested to associate the fulminate, include fulminate pathogen as one of the pathogen in comparative studies. So with respect to this, we are studying all that cross-infection studies of that millet pathogens which are infecting fulminate, fast-tail millet. Even recently this last disease also increasing in other crops, something like banana millet and regular and specific locations. So here the results show that so this is done only for regular isolates. First we have identified regular isolates and then we have carried out these cross-infection studies. So regular is able to infect master millet, banana millet, brown crab, and even pearl millet also. So whereas the particular result is able to improve mobility tasks among the study, that is one is by mention to millet, it is able to infect master millet as well as brown millet. So master millet, so we are able to infect mention to master millet, infected banana millet, brown millet, and even other millets. So this we have other publication studies in plant pathology,
Society for Plant Communication, so the data is available only. Then coming to the plant methodology approaches and achievements. So these are the applications. So we have regular centers and two one centers which are located across the 7 states. And we have the overall program, that's a number of projects which are aligned across the different centers. So we have 120 trials, of which 116 trials were conducted across the locations, which is accounted for 95.08%. Then coming to the first year, we gave these surveillance for the important disease. So here the objective is to see the response of new disease varieties under the different conditions, under the different climatic situations. So we have some of the common diseases which are across the locations, or something like a food and septosomal for compared to the only specific locations. So when come to the scenario of small diseases, especially disease with respect to in the harmless way. So here the lead was the maximum incidence of the lead was recorded at Karnataka, Mangalore, the surrounding district, or the Ranitore, and low to medium moderate incidence in other states was observed with respect to lead was. Then the moderate high incidence of lead was recorded at the Mangalore, and Bangalore, and Ranitore Center. Disease resistance. So in every also had similar kind of disease scenario. So here the activities were created in live locations and medicine and they also multiple disease resistance. So in another point is in hospital activity. So there are the common diseases which are we have observed as a last burst and innovations and prostate. And here the last low market incidence of Ebola was recorded at Bengaluru, Mandya, incidence of last of Mandya and Ranchi, and low market incidence of rest was recorded at Bengaluru and Vijayanagar, and later incidence of rest at Mandya Center, and one decline is also the high incidence of Ranchi and Vijayanagar. So here we have a increase in 6 locations. So each promising and 4 members also multiple disease resistance. So another problem is it's area of alternative so this is also one of the diseases reported recently from our from India and the particular so it's the one disease and another is the kind of it's only compared to a few locations. So in LAVT also we are not that much disease problem. So here 21 entries were oriented in 7 locations. So almost all the entries have shown a resistant to automated blight and bone spot and the field conditions. So in so this is one of the important diseases, that's the X-Lux and Bender. So these are the 2 major diseases. And the low to market incidence of X-Lux was recorded at Bengaluru, Jagdalpur and low to moderate and brown spot at Bengaluru, Nandyal and Mandya Center, and moderate to high incidence of landed pain was recorded at Jagdalpur, Ranchi and Vijayanagar. So here we have evaluated 13 entries in 8 locations, and 7 were promising and 4 were from multiple disease resistance. So here also we are having only big problems with leaf blight and banded blight. So here 40 entries are evaluated in 6 locations. And 8 were promising and 4 entries are for multiple disease resistant. So another crop that is barley millet. So here it is grain plant, eggs plant, leaf blight and major farmers. So here the market incidence of rice among Bengaluru and market incidence of agriculture in India. And also market incidence of Ranchi and Vijayanagar. So here this 13 entries are related to 8 locations of promising and 3 improves of multiple disease resistance. So another one is was recently introduced. So here we have disease, one is the leaf blight, another one is the rust. So this leaf blight was also reported as a first report from India, was produced in the blind disease, produced by ABS and these 2 diseases are coming in this so here, incidence of the was recorded in almost all the tested center, and even all the incidence of rust was and Bengaluru, and even Bangalore was specific location or minute later, all the incidents. So when these evaluated in 8 locations, so they were promising and we also multiple disease resistant. So in addition to this, we have another set of that is innocent. So we have to share somebody, so we have medicine for given entries, 8 locations, in Guru, 8 entries in 5 locations, including 11 entries in 6 locations, and Margaret 27 locations, total 78 entries for texture across the locations based on this location, and promising entries for identified in the innocent also. Now coming to another set of is immunity, this we are going to run through. So here we have we have selected some of the policy lines and we have recommended these entries and within the environmental condition from the 22 and 23 and this year we are controlling this there. So how we stress here about entries which are home resistant to the almost days of last. So these are also the artificial conditions as well as features say we are measured these and we also exposed to 4 weeks of pathogens which are connected to different locations, that is something like the plains, hilly areas and coastal areas. There are entries which are widely that can be used for the program. So another immunity, so this also we are conducting from the last 3-4 years. So in the first year, so 300 entries, every year we are getting ready, eliminating the entries which are found susceptible, and for the last year we have concluded 34 resistant for the blast as well as rust, and this year also we are repeating this year and we are going to conclude this in the next year. So some of the entries they have done very promising, even for both blast as well as rust. We can see in the photographs, but so this we want to repeat once again, and we are going to conclude in the next year. So this is all about the second, third thematic is disease management. So this is management of water rust with the vitamins and fungicides, and we are having vital agents and services. So as I told, this is also one of the important disease, different combinations. So here among the main treatments, C treatment to the microbial consortia and development fertility, and saying the some of the significant microbial consortia and development. So this is both the centers we have touched, and in some of the centers we are not have so we have to bring this fire. So specifically also on the basics of this C treatment in the microbial consortia and but we are going to really in the next season. So we are also set up assessment Millets, and so here we conducted last 3 years. So the Millets and similarly other Millets is master so we have conducted for the last 2 years. All these 7 experiments we have concluded with this year with this information data. So in other words, the yield so just these are the exhibits we have collected. So we have and most of us have indicated we have some good publications that some and also there are some pathogenic groups, and on the 6th area this is a genomic database. So here also we have done 6 genomic data is there, and we have to work out and we have to come. So in the last year we have one that is a major salami, that's we have collected 7 isolates, and we have sequenced and identified virulent and old genome, and we have compared. So we have found genetically 6 clear indicators, 6 genetically distinct, and it was found cluster before Egyptian salami, which is causing seed plant in rice. That's EG1 group, and we have nearly about 19 million reads with 100 X coverage, and this assembly is almost compared with it. And we have also made comparative genomics with respect to this rhizophonase salami impact in farmland millet, rice, maize, and soybean. So it was similar with that of rice and maize and the soybean. You can see that there are some of the genes are unique, and there are some unique proteins which are 24 that's exclusively present in case of farmland millet. And we also looked at some unique protein, especially with respect to the cutinase. We all know that it's one of the important enzymes or protein which is plays important role in the breaking to cuticle, and plays important role in the most pathogen compatible reactions. And here we find some interesting thing that as you know that this cutinase generally composed of around which is a division of residues. So it will reach the and also this is millets, and we also compared with so there are some unique proteins and common proteins, and we need to get to understand the role of each one. And there are some other students who sequences over 600 these sequences for publications. So this is the centers 1 and 2. And there are some other publications, awards, and other activities with respect to international this is a technical program, and this is our, so this is our team for the next academic year. Thank you, sir. So this person is team with respect to the genomics, person is speaking. Thank you, sir.
Thank you, Dr. Balama. Any question from audience side? these are micro introduced. They have recently begun a very good study. If you have any questions, please just raise the hand and ask you. Okay. From my side, I have one correction. Now, as far as the co-chair is concerned, I have told you that Dr. Giri Reddy was working as a co-chairman, but Dr. Reddy Kumar, Professor of Plant Pathology, is working as a co-chairman, and I am handing mic for his remark to you.
Thank you, sir. At the outset, it's my pleasure to be part of this annual meet. And first, I am very thankful to most respected DDG sir, Dr. Sharma sir, and Director IAMR, Dr. Tara Satidevi garu, other delegates, and our Honorable VC Madam, Dr. Sharada Jayalishmi garu, for giving me the opportunity to be co-chair of the session. And so there are excellent presentations made by Dr. Prasa and Dr. Palana sir. So as far as those, so very innovative approaches were made as far as the millet crops concerned, using different approaches like microbial consortia, and also the using some of the nano formulations. Very quality slides and good presentation was made. And so as far as the second presentation goes, Dr. Palana sir, very excellent presentation, and incorporating all kind of newly available technologies like whole genome sequencing, which is gaining a lot of importance at present. So if you look at as far as the fungal pathogens and microbial diversity concerned, this part is taking a lot of importance as far as the planetary side is concerned, and so very good studies were made. Thank you very much, sir, for your elaborate and detailed presentation and for quality studies. Thank you very much. Thank you all for giving me the opportunity. Thank you once again.
So early morning, Dr. T.R. Sharma, DDG Crop Science, who happened to be, I think, plant pathologist also. He has told about the races. It is one of the most important things, because in every pathogen there are races. Whenever I am working on as a chickpea pathologist, whenever we send the material for the multi-location trial to everywhere, every centers, we found that certain genotypes, they are showing the resistance at certain places, and certain genotypes, they are showing the susceptibility at certain places. It means there exists the races in the pathogen, and accordingly we can go for the gene requirement. Now we have given an excellent example of coccidia path, where according to coccidia path, how we are as per the biotype, we are dividing each genes so that the pathogen pressure can be reduced as well as the toxicity can be reduced. Such criteria should be adopted. Now in almost all SAU we have the AICRP system working, and most of the AICRP workers, they are getting student PG or PhD students, and it is my sincere request to all of them that whatever the pathogen in their area, they should go for their genome mapping, their street building, and at the same time they try to develop the prognostic model, because prognostic model is the most important thing. Until and unless we develop a computer simulated prognostic model which is linked with the IME data, which is long-term and short-term forecast, such forecasting is in place. the proposed spray or proprietary mediated technology definitely reduce the chances of occurrence of the pathogen, and there may be increase in yield. And then I must congratulate both the presenter for their excellent presentation, and I co-chairman also Dr. Kumar and Dr. Deputy also at the same time to the audience listening to all of them very patiently. Thank you very much.
Thank you very much, sir. Thank you very much, Dr. S.S. Mane, Dean PDKV Akola, and our PAMC member for sharing this session, important session on crop protection, that is pathology. Thank you for your remarks too. And as exactly you told, forecasting for disease and diseases is very important so that we can have the early identification and early management. I thank Dr. Professor Reddy Kumar, Professor of Plant Pathology, to be there as co-chair and taking over all those proceedings here. And I thank Dr. I.K. Das and Dr. K.B. Palana for the presentations. I request Dr. C. Tara Satyavati Madam, Director ICR to come onto the stage to felicitate the chairman and the co-chairman of this session on plant pathology. To the chairman, Dr. S.S. Mane, Dean PDKV Akola, and the PAMC member. The co-chairman is Professor Reddy Kumar, Professor of Plant Pathology. Thank you very much, madam. Thank you, sir. We will be there for a group photo. Okay. Friends, with this session, we are finishing the technical sessions for today's program, and we are breaking for tea break now. And we will be going for the session 3, that is VIC meeting at Institute of Frontier Technologies at RARS. So the members of VIC are requested to go to Institute of Frontier Technologies, RARS. So only VIC members. And all others can break for tea and come back here for cultural program by 6:15. So it will be 6:30. So only the VIC members are requested to go to Institute of Frontier Technologies, RARS, and the other members and delegates are breaking for tea. Thank you.
వీడు చేసే పిల్లలు కొనుక్కోవాడు ప్రతాప్ ఆ కమరా లైవ్ ఇచ్చా
All the VIC members and
ఇదంతా
all the VIC members and VIC of the program are requested to go to Institute of Frontier Technology, RARS.
ఏమన్నా మెసేజ్ రాలేదు నాకు నంబర్ చెప్పు నంబర్ అది పక్కన బిల్డింగ్ పోవాలంట నంబర్ చెప్పారా ఎనిమిది సున్నా సున్నా 8 0 2